Go.
Hi, welcome everybody to the second debate with Catz and Standing for Truth about all kinds of important stuff that I don't really understand. Ancestry, is it universal or shared, young or old? Earth, I suppose. Right, guys?
Yeah.
Universal and separate, but they both start with an S, so close enough.
Oh, yeah, sorry.
No, you're doing good.
I have it still the wrong title. I got the thumbnail right, though, this time. So,
No
anyways,
worries.
with me, as always, we have Cliff,
Hey guys, how you doing?
and Robert. Robert,
Hello,
you
everyone.
gonna say- there you go.
How's everybody doing?
And of course, the stars for the whatever long it's going to take. Conspiracy Cats,
Hello. How we doing?
our hero, and Standing for Truth, our guest.
Hello, good to be here. I'm excited. Thanks for
Guys,
inviting me yet again.
What-
you know you made it when Steven Steen is at your debate.
I know, right? Hey, hi, Steven. Good to see you. It's been a while.
Steven, can I get an
All
autograph?
So we have a few super chats I should read. Before we even started, we got one from Brainless Steel for €5 that I can't get back to, but he didn't know if he was going to be here or not. So I'm glad you're here, Brainless. Really glad you're here. Then let's see. We came to Anamorphic Mind for $2. Wait, this isn't Pornhub? Cute, real cute. John Rapp for $2 Australian. Okay, have coffee and cigs ready when you are. Me too. I made my coffee before we started. Good one. And I think Steven Steen. Congrats, Stand for Truth, on your easy win. Yeah,
I paid him to say that.
you can't get me mad 'cause I know you, dude. All right, so Snark for €2. Let's get this thing started, and we will. Who did we decide going first? Standing?
Standing.
Yes.
All right.
Catz went first last time, so it's only fair that I go first this time. So
Great. Well, before we start, all super chats will be read. If you have a question, you can send it to super chat. If not, at me. What we're gonna do is question and answer afterwards. We're gonna start with the intro speeching thingy for 10 minutes, then rebuttal for 5, and then discussion. I doubt I'm gonna have much moderating duty because these guys are real cool. But hopefully, somebody will step out of line and I can smack them. All right. Ready when you are, Standing for Truth.
awesome. Okay, you can hear me okay? Audio sounds good?
Loud and clear.
Okay, yeah. Before I start my timer, I just want to thank Catz for agreeing to do a second debate. I really enjoyed the first debate, and it was great on this channel. The host, the moderators were awesome, were kind, were fair. So I'm really looking forward to round 2. So let's get started. I'll start my timer here. I've got 10 minutes, so here we go. 10 minutes. So debate on ancestry, universal or independent. Today we are debating the origin of species and the question of ancestry. When it comes to ancestry, the best place to look is genetics. Genetics can tell us whether all life on Earth is related or whether the Genesis account of Adam and Eve and special creation is true. It's in our DNA where the question regarding origins is answered. Sperm and egg do not pass on a fossil or a bone. They do not pass on geography or a rock. They pass on genetics and traits. The answer, I hear a bit of an...
Can I interrupt? You got an audio.
Yeah.
I'll pause the timer real quick. Is that coming from my end or? I'll pause the timer real quick.
Yeah, I'm
Yeah,
hearing
somebody's screen is playing back.
Yeah, my screens are all shut off, but I can definitely hear the echo there from
Does anybody
myself.
have the audio on for theirs?
I don't hear it now. I don't hear it now.
Mine's off, yeah.
All right, let's try it again then.
Okay, let me...
I'll wait
Okay.
till you start. I'll start the timer again.
No problem. I'll skip back just 10 seconds just so I know where...
Absolutely.
You just started, so if you want to start over and get your flow going again, feel free.
Sure, yeah. Okay, good idea.
10 minutes.
10
Starting
minutes.
now.
Go.
Awesome. Okay, so today we're debating the origin of species and the question of ancestry. When it comes to ancestry, the best place to look is genetics. Genetics can tell us whether all life on Earth is related or whether the Genesis account of Adam and Eve and special creation is true. It's in our DNA where the question regarding origins is answered. Sperm and egg do not pass on a fossil or a bone. They don't pass on geography or rock. They pass on genetics and traits. To answer some of the important questions on ancestry or find out the history of humanity and discover the origin of species, this is where we must look. What about fossils? Well, we can look at dogs as a basic example as to why genetics is so critical in determining what is related and what is not related. Dogs of all types make it clear that morphology, anatomy, and bones in the dirt can oftentimes fail us because there is often more diversity within a species than between the species. This makes it difficult to classify animals by their physical traits alone. Therefore, the best way to determine ancestry is by looking at the genetics and that which is passed on sperm and egg. I predict conspiracy cats here will probably look to rocks to determine a deep time history regarding humanity. For example, he may look to isochron dating. Well, isochron dating analyzes 4 or more samples from the same rock unit. The RATE team, consisting of many PhDs, PhD geologists, PhD geophysicists, have discovered discordant dates even with the isochron method. So let's get right into the facts then. Creationists are the ones making testable predictions on DNA function, mutation rates, speciation rates, and more. The genetic structure in humans today speaks to us of a literal Adam and Eve. I'm not trying to sneak up on Catz here. I thoroughly enjoyed our first debate, and many of the questions I am asking here today were either not answered sufficiently or simply not answered due to time constraints, which is why we're having the second debate. How does Catz, for example, explain the mitochondrial DNA and the Y chromosome data that based on the empirical method tells us we come not from just a small founding population, but a population of just 2, Adam and Eve, 6,000 years ago? Conspiracy Catz is faced here today with an extremely serious challenge. That challenge involves pedigree-based human mitochondrial DNA mutation rate studies. The reason for this is because pedigree-based studies and the empirical method rejects deep time evolution and universal common ancestry. The best tools to determine what is related and what is not related are molecular clocks and DNA function. Not a bone like Catz may look to, not a rock, not geography. Yes, these are indirect ways to determine ancestry, but the only direct way is in genetics, and molecular clock data both confirm a young time scale and refute an old origin of species. Evolutionists look to time dependency to explain away the data. They have suggested that the mitochondrial DNA clock ticks at different rates at different points in history. Evolutionists would have us believe that mutation rates have been significantly slower in the past. For this explanation to be scientific or even reasonable, Catz here must make testable prediction. And this is what I am going to request he answer in the discussion portion. If he disagrees with the Eve date derived from straightforward mitochondrial DNA coalescence equations, he needs to make testable predictions. When does the molecular clock speed up and slow down, for example? Can Catz here predict when the mutation rate accelerates and when it does not? Or is time dependency a rescue device that evolutionists are forced to invoke to explain away the observable data? When we focus on the variation in the Y chromosome in the world today, for example, we see that Y chromosomes have very little variation. This indicates that we came from a common ancestor in the not so distant past. A new research paper here, you can see it on the screen, by Dr. Nathaniel Jensen, confirms that we only observe approximately 4,500 years worth of mutation accumulation in the paternal ancestry that is contained in the record of this human Y chromosome. Subsequent papers also confirm testable predictions on the history of civilization found in the Y chromosome. How does Catz deal with this? What about the new genetic boundary study that concluded roughly 90% of all animal life was roughly the same very recent age? Once again, evolutionists resort to post hoc, ad hoc rescue devices to explain away the very clear conclusions that are perfectly consistent with a recent origin of species. I'm looking for testable predictions because the best evidence for universal common ancestry, which Catz is here defending, has been overturned. The entire junk DNA paradigm has been overturned. The chromosome 2 fusion has been overturned. We discussed that in great detail last debate. We can get into that briefly a little bit more here because I am curious for the fact that chimps have very distinctive and exceptionally large satellite sequences at the ends of their chromosomes. Where are these large age-specific satellite sequences in the reputed fusion site? Here's a paper that confirms what I'm saying. Catz did provide answers to some of the problems for the chromosomal 2 fusion. And although I feel the answers were insufficient, the question regarding the ape-specific satellite sequences is a question I'd like to hear an answer to. ERVs, ALUs, and other classes of retrotransposons, and even pseudogenes are consistent with the created heterozygosity model. This hypothesis would suggest that Adam and Eve were created with pre-existing functional DNA differences. This would apply universally across the biblical kinds, and this makes sense both scientifically and theologically. This hypothesis makes extremely specific predictions on DNA function. The evidence is clear. These are functional DNA elements important to our genome. Since we know that ERVs and other classes of retrotransposons accomplish many crucial functions in regulating gene expression, differentiation, development, and even cell stress responses, here are numerous papers confirming the incredible functionality of these DNA elements. Can Catz here provide any empirical evidence, maybe a technical paper, that actually demonstrates a non-functional DNA element such an ERV, pseudogene, or ALU going from non-functional to something critically functional in the genome? And I'm not looking at rescue devices and post hoc answers involving co-option. And well, not all of them are functional. I want to see real evidence as to why we should believe these are not created units of DNA function. We have preliminary evidence for genome-wide functionality. Once again, this is a direct prediction of the created heterozygosity model. Paper after paper after paper, as you can see here. Can Catz refute this? For example, nuclear protein multifunctionality. This is exactly what we predict for these highly conserved ATP synthase proteins, such as cytochrome C. Can Catz make better predictions, for example? Also, orphan genes show limited ancestry. How does Catz explain this incredible class of taxonomically restricted and essential genes? Orphan genes are exactly what we would expect given separate ancestry, which is what I am here to defend. Maybe Catz can start off the discussion by answering this or any one of these extremely important questions. A couple more questions I would love to see answered. How does Catz explain the incredible dissimilarity, you can see the paper here, between chimp and human Y chromosomes? How does Catz explain the low genetic diversity in human beings? The Bible tells us that God created two humans, Adam and Eve. Therefore, a direct expectation, if this were true, would be a low genetic diversity in humans. And this is exactly what we find. Evolutionists were shocked by this, which is why they invented the hypothetical out-of-Africa population bottleneck to explain this low genetic diversity. If Catz is willing to answer this question, I'd love to discuss the out-of-Africa scenario of human evolution. How much time do I have, guys?
You have,
You have 2
minutes, 15 seconds. I was about to give you the 2 minutes.
Awesome, thank you. What should we discuss tonight? We need to discuss the differentiating evidence to determine which model is strongest, creation or evolution. There are many lines of evidence that are agnostic to the debate. For example, we often hear evolutionists point to homology, the shared structures seen in the biological world. But we know human engineers design in homologous patterns. Across the globe, we see shared designs and even shared blueprints. They often point to the so-called existence of transitional forms. Well, think of a military vehicle that blends the features of both a land vehicle and a vehicle built for the ocean. For example, as you can see here, an amphibious assault vehicle. This would be the perfect example of a transitional form according to Catz here, when in fact we know this is not the case. This amphibious assault vehicle was built and designed this way. What about the groups within groups patterns evolutionists point to as evidence for descent with modification? Nested hierarchies in DNA, anatomy, and physiology are also predicted by creationists based on the design model. Nested hierarchies are the products of design, as you can see here. Are these patterns reflective of function and design, or are these patterns reflective of descent? These lines of evidence that Catz and evolutionists typically look to here turns as a way to argue against a literal Adam and Eve and limited ancestry. You have far too many explanations that can be invoked. Therefore, they are agnostic to the debate as they cannot differentiate between the models. These lines of evidence do not work since both models fit the data. It's a lot like saying the sky is blue or the earth is a sphere. Therefore, creation is wrong. Excuse me? Both models can explain the shape of the earth and the blueness of the sky. What does this have to do with the debate? There is a whole lot of explanations that can explain the data. Just two lines of evidence that can differentiate between the models, and I hope Catz here is not afraid to discuss, has to do with the molecular clocks I described above.
You have 30 seconds, by the way.
Awesome. Okay. Everything we see today points to the death and destruction of a once perfect creation. This includes our genome. What type of selection can be presented that will select away so many deleterious mutations that are pouring into our genetics and degenerating our information systems? Almost done here. Catz provided an answer in our first debate involving bacteria. This was a very unsatisfying answer. Will Catz admit humans are presently degenerating, or will he actually assert humans are getting better? Most mutations are unselectable and invisible to selectors. And yeah, so there's lots of questions here that we can discuss in the rebuttal round and discussion portion. I look forward to these portions of the debate, and thank you so much for listening.
Thank you. That was really interesting.
Very nice.
Okay, Catz, over to you. Can you follow that one up?
You're muted, Catz.
You're muted, Catz.
Okay.
You know, while I was muted, I totally disproved creationism. So my job's done for tonight. If you missed
yeah,
it,
there you go. All done. Oh, it's over.
I was gonna say, standing for truth. Would it be okay if before I start my time, if I just fire out just a 1 minute little message? Is that okay? It'd be very, very quick. I think debates like this I really, really enjoy, and I enjoy them because they give me the chance to learn new stuff. Okay. And not more so in the past few weeks. There's somebody in the chat watching now who I'm going to forget as I'm going through the stuff that I'm going to talk about tonight. I'm going to forget every time something comes up, which will be 95% of the time, 99% of the time. There's somebody in the chat called Anthony Purcell, who has helped me massively. He's a proper scientist, not like me. So, and I've learned so much from him. So I just want to say thank you, because if I do forget to credit you, it's just because it's the heat of the moment and me talking. So almost everything that comes out of my mouth will be from that man, and I do hope that I can do him justice. So that's just all I wanted to say.
We love Anthony here.
We had a great conversation between Marvel and Anthony with the other night on stream, actually. So yeah.
He's a really, really knowledgeable guy.
Yeah.
So sorry, I've lost you there. Are we good to go?
Yeah.
Are you ready for it?
Yeah. So essentially, can we start with just my first slide on the board? Is that okay, please, Robert?
There you go.
Let me know when the clock starts, and I shall begin.
Whenever you're ready, you start talking, and then I'll start it.
Right. Okay, let's begin. So the last debate we had was about genetics and how it supported evolution. This one is about shared ancestry and the age. Are we old? Are our species old? Our species young? And in the last debate on genetics, we come to an agreement that there's nothing about genetics that defies evolution. Genetics doesn't in any way contradict evolution. Standing for Truth made the claim that he thinks it also fits creation. Okay, that's fine, but there was no evidence presented at all that current genetics defies evolution. So I'm focusing this debate a little bit more on the age side. There's two titles. So this it kind of is our ancestry old? Is it young? And when we talk about creationism, old and young is defined by 6,000 years old. That's the age of the earth. 4,000 years old would be anybody who's lived after the flood, when people started to die, etc. And I think when we start looking at it that way, there are massive, massive, massive flaws for creation, which unfortunately, standing truth, I'm about to expose for you. Can I have slide? Sorry, we'll have slide 2. I'll talk about that one later on. Now, Standing for Truth did bring up isochron dating, and I thought he would, and he was quick to get in that isochron dating he felt wasn't reliable. And we can talk about that later on because it's perfectly reliable. But here are some things that he just can't refute. Here is a study that you can see on the screen. Hopefully you've all got that, from 2004. Now, in 2004, somebody took 50, and can we just skip onto the next slide as well, please? In this study, you can see the map there on the next slide. There are 57 locations on Earth, 57. And these 57 locations all had a core drilled from those 57 locations. And if you move on to the next slide, which would be slide 4, Robert, we can see that the people who took these 57 different samples are all listed here underneath figure 2. And it's not these are very, very independent studies. And studies and dating techniques were applied to these samples to build up a record of historic oceanic volume and temperature. Now, if Standing for Truth is correct, and that our dating techniques are flawed, when we took these 57 completely independent measurements from all over the world, and then we compared them together, we would not expect to see what you're about to see if we look at slide 5. Is slide 5 on the screen, Robert? Because there's a bit of a lag. Robert.
Slide 5 is showing, yes, sir.
Oh, brilliant. So this here is all of those fluctuations vertically aligned. Now, looking at it at slide 5 as we've got it there, it doesn't really show the detail I want to show. So if we zoom into one aspect of it and we look at slide 6, so we can go to slide 6 now, please.
6 is up.
Then we can see an unbelievable, almost identical pattern in every one of the, and this paper, anybody, you know, I'll put a link to it, and if Anthony was okay with that, I'll put a link to that in the description. So we have 57 independent points all over the globe that have been drilled out by different independent scientists, and these dating techniques to measure the volume of the ocean and the temperature of the ocean over hundreds of thousands of years have been applied. And if these dating techniques were flawed, how on earth is it possible that they all come back giving exactly the same result? How is that possible? Now, I'm going to really hammer this point home in the back and forth in the questioning. How is it possible that 57 different experiments, if you like, individual experiments, all yield the same result? But it doesn't stop there. Okay, it doesn't stop there. If we go on to the next slide, we can talk about dating ice cores. Now, you might say, I've heard creationists say that when you date an ice core, are we on slide 7, Robbie? Yeah. When we date an ice core, all we do with the ice core is we count the number of layers and then we derive an age from that. But that's not true. If we go on to slide 8, there is a whole multitude, again, of different techniques, absolutely different techniques that are going to be used to date the ice core. So we can count the annual layers. We can look at atom trap trace analysis, which I believe is to do with krypton in the air. We can look at uranium decay in dust grains, and obviously they're both examples of radiometric testing, but they're different methods, they're different isotopes. We can look at greenhouse gases, etc. And if we can skip on 2 slides, is that okay? Just 2 slides to slide 10.
Slide 10 is up.
So slide 10, the graph. Here we've got yet another study, okay? Yet another study with a massive range of different techniques. And I really want this to sink in, Stand for, because this really just destroys your argument before the debate begins, I feel. We have this huge range of data going back hundreds of thousands of years, again, from, if we look at Epica, the blue line at the top, that's one location. In Stock is another location in Antarctica. They have been independently tested, again, for temperature, like temperature records going back over hundreds of thousands of years. And again, we get almost an identical image using these different techniques on the same, different techniques in different places, but testing for the same result, the temperature going back over 100,000 years. If these techniques are flawed, how is that possible? That is not possible, as the previous example I've just given you is not possible if the techniques are flawed and are giving random results. Now, what's even worse for you is if you look at that red line at the bottom, which talks about the volume of ice over time, that's measured by using oxygen that's linked to like microscopic marine fossils, so life, okay? We have looked at life over hundreds of thousands of years, not over 4,000 years or 6,000 years, hundreds of thousands of years, used a completely different technique to measure the ice volume. And you can see exactly the same pattern as we have for those 2 independent locations. So you've got 2 studies, real world studies published in proper journals right there that show a wide range of different techniques giving results that haven't, the coincidence would be unbelievable. It's just laughable to think that that is down to coincidence, going back hundreds of thousands of years. So to say, I don't think radiocarbon dating works, or I don't think isochron dating works, or I don't think uranium thorium decay works, or I don't think thermoluminescent dating works, whatever, when we combine these together, we should be getting different results if they don't work. And I think I've shown you that we do get the same result. Now, if we move forward to slide 13, again, this is where it becomes incredibly unfortunate for creation, because when we're talking about is our ancestry young or old, and ancestry is defined by creationism to be old, has to be over 4,000 years, because there was only Noah and his family who stepped off the ark. If I find this, are we on 13? Sorry, Robert.
Yes, sir.
Brilliant. Thank you, sir. If I find this piece of pottery, just like the creationists would say, there has to be a designer, and that designer will be human. So all I have to prove is that that piece of pottery is over 4,000 years old, and we have civilization pre-flood. And again, creationism would have no answer to that. And what would happen if I go on, and I will do, and talk about a wide range of dating techniques that all corroborate each other without contradiction?
You got about 2 minutes, by the way.
2 minutes. Brilliant. Thank you. I'll speed this up. Next slide, please. Welcome to the world of thermoluminescent dating. And we use this to date pottery. If I just move on one more, please. Now, thermoluminescent dating standard 2 doesn't rely on ratios of isotopes. Thermoluminescent dating, and I know I'm not got much time, but we can go to slide 17, please. Thermoluminescent dating relies on radiation, background radiation, and internal radiation, affecting the electrons in a lattice so that they become excited and they jump away and they get trapped in what we call electron traps or imperfections in the lattice. Now, as long as that pottery is exposed to the sunlight, these electrons get to jump back down again, and the clock is reset, it's bleached. But as soon as there's an event such as burial or this pottery being put in the pyramid, the clock starts ticking and that charge starts building up. All right, if we go to slide 19, we can see that what essentially this means is when we look at how much, and we measure that charge by how much light is given off when we heat it, essentially, that because the clock keeps getting reset every time it's exposed to light, we're not actually looking at how old the pottery is. We're looking at the last time it was exposed to daylight, and it only takes about up to 100 seconds. Now, if we go on to the next slide, slide 20, if we want to talk about contamination, contamination actually makes it look younger. Anything that contains water absorbs these electrons as they make their way to those electron traps, and that actually reduces the charge. So thermoluminescent contamination makes it look younger. If you go on to 21, you might say, well, what if there was some kind of catastrophic event in history that meant there was loads of radiation on these buried items all in one go, and it suddenly made them look older? Well, it turns out that pottery fraudsters...
We're at time. We're at time, by the way. Yeah.
Darn it.
Just you can take a minute to close out, or take a couple seconds.
I'll be very, very quick there. It turns out that people who try and fake ancient pottery bombard the pottery with radiation to falsify this charge, to make it look like it's been buried for a lot longer than it has. And the answer to that is on slide 22. I'll be 20 seconds. Now, if there had been some kind of catastrophic biblical event that somehow had gone unnoticed by everybody else, but bombarded all the pottery in the world, we still would notice, because when people try and fake this for real, it turns out, and I've underlined the relevant bit there, the size grains in the clay will have different doses unless we've had one massive dose given in one go. They will all show the same charge if it's been artificially irradiated or heavily irradiated in some kind of unnatural event. So we can spot the frauds. So what you've got there is, I'll take more than my time, but I think I've shown there that creation doesn't answer any of those questions.
All right, Catz went about a half a minute over. I'll give you that extra time. Standing for truth on your rebuttal.
Awesome. Okay, thanks so much. Well, I appreciate that opening there, Catz. Catz focused entirely on the age of the Earth and not on the age of the species and the age of humans. So he's failed right off the bat, unfortunately. The pottery made right after the flood would have had high amounts of radiometric elements in it that quickly decayed, like all the other elements did in the stable isotopes. And obviously, if radioisotope decay was accelerated, say during the Genesis flood, then all of these radioisotope decay clocks, for example, could never actually be relied upon when they date rocks of, say, millions and billions of years old. And there's actually several independent, he talks about independent lines of evidence. Well, there's many independent lines of irrefutable evidence that accelerated nuclear decay did occur in the past. For example, we have evidence in the rocks themselves involving helium, fission tracks, and radio halos. So this is observable evidence in the rocks that nuclear decay has occurred. All of these dating methods he looks to, they're just relative. They're not absolute. Genetics does disprove evolutionary theory, though. I'm not sure if he understands the point of the entire last debate, that DNA function and predictions, for example, would overturn the nested hierarchies that he looks to, and that would demonstrate that those hierarchies are there by design and not there by descent. So, for example, just a few lines of direct evidence, not these indirect lines of evidence, are orphan genes. I'd like to hear an explanation from Catz regarding orphan genes, molecular clocks, for example, Y chromosome and mitochondrial DNA, genetic degeneration. The only argument he had last time about genetic degeneration was bacteria. Let's get into humans. Are humans getting any better? He talks about these independent measures. No, they collaborate. They calibrate the data all the time, especially on the molecular clocks. Catz assumes away a global flood and a special creation event, and the ice cores, for example, and everything else he looked to, uniformitarian assumptions. But the question is, why do so many independent molecular clock studies converge on a young date? I want an answer to this. I didn't get any answers in our first debate. So it looks like he's thinking because civilization says that they're older than 4,000 years, or because some radiometric dating says that they are older than 4,000 years, then it must be true. These are all indirect lines of evidence, because genetics, that's what's inherited sperm and egg, and genetics disproves everything he's saying. The direct evidence suggests that humanity is no older than 6,000 years. Let's get into the actual species, since the debate is origin of species and the question of ancestry. Why are there so few species today, if deep time evolution is true? And we can go over a number of examples with the number of species of snakes, lizards, birds. We can discuss that in the discussion portion, because it's a fatal blow to Catz' position here. Even the chart he showed in the beginning, you can see a constant temperature in the ocean, for example, which is exactly what our model says the pre-flood world was like. Then the flood hit and chaos began, and this is why the ice age shows fluctuation. So nothing he showed, for one, proves his model of ancestry, since the molecular clock data suggests otherwise. And his lines of evidence assume uniformitarianism, for example, and assume away a global flood. And as I showed in the beginning, for example, even the isochron method, which they believe is immune to some of the assumptions and some of the claims of constant rates, for example, even they come back with disconcordant dates. There's a huge window of error when it comes to these dating methods. So I'm really interested to get into the differentiating lines of evidence, especially in genetics, that we didn't really get to cover in our first debate. So we can even get into lines of evidence that I'd be interested to see how he can explain in the Earth today, to show that the Earth is young, for example, like coal and diamond are very frequently found to contain carbon-14, even though carbon-14 decays so fast that it should be undetectable in far less than 1 million years. He can't explain that away with contamination, of course. The magnetic field of the Earth is decaying. Comets are losing so much material from passing the sun that they should be non-existent in an old universe. But we can discuss all of these things, of course. The catastrophic plate tectonics model, runaway subduction friction would wreak enough havoc to accelerate nuclear decay in the rock. So these dating methods are all relative. They're not absolute. They can come back as millions or billions of years, when in fact they're only thousands of years old. But we need to get to the direct lines of evidence to demonstrate and to answer the question today regarding ancestry and the origin of species. So it's detrimental that Catz answers the data on molecular clocks, genetic degeneration. What about the Y chromosome dissimilarity between humans and chimps? If chimps are supposedly our most recent common ancestor, why the huge difference, especially if the Y chromosome is immune to recombination, for example. So there's just so many discrepancies. While Catz here wants to look to pottery, ice cores, he wants to look to rocks, all of these indirect lines of evidence that assume uniformitarianism, assume away a global flood, assume away a special creation event. Let's look to that which is inherited sperm and egg, that which we can look at and track back, and only goes back 6,000 years, 4,500 years to Y chromosome Noah, for example. So I feel like I addressed all of his argument in the opening. He didn't really present much regarding ancestry, but is that time?
Mm-hmm.
Okay, well, thank you so much. And Catz, go ahead.
Cool. How long, just so I can plan this out, how long have I got? 5 minutes? Okay, so let's begin now. So this is a rebuttal, so I've got to touch on what you said in your opening statement. You talked about ice cron dating and how it was flawed, except you didn't give any evidence or any kind of papers or citations that show it's flawed, or an explanation as to how it's flawed. So I'm just going to move on from that one. Mitochondrial DNA, can I have a slide 120? Sorry, slide 122, please. So you mentioned Adam, Y chromosome, Adam. Listen, this took me about 5 minutes just to go and look at a bunch of papers that were not in creationist journals, but scientifically peer-reviewed journals, and all of them completely disagree with what you just said. And you've also got to remember that Y chromosome, well, mitochondrial leaf certainly is a different, that person can change depending on when, through time, as different lines die out, that person's going to change. But none of what you have said is reflected in any mainstream science whatsoever. So again, without an actual paper, an author for that, it just doesn't marry up. It's just, if you like me, just saying, I don't believe it. It just doesn't marry up. We talked about satellite sequences. If I can have slide 83, please. You talked about the satellite sequences and the chromosome 2 fusion. Well, here is the chromosome 2 fusion point on, was it 83? Have I got that right? No, I haven't. Oh, I've lost it. Where's my picture of the chromosome?
Space comma.
I've got to find this slide.
Okay, I'll pause.
I will get there. Sorry, I've got myself a little bit muddled up. Right, I will show it later on, but I do have the paper here with the picture of the chromosome of the fusion point. And when you're talking about satellite DNA, what you've got to remember is that the mutation rate, when we get to telomeres and telomerik satellite DNA, those are the parts of the chromosomes that are constantly being replaced, and they are much less affected by mutation because they are constantly being renewed with telomerase enzyme. When those parts of the DNA are in the center at the centromere fusion region, they're not protected from mutation anymore. So what you might not recognize as satellite DNA is actually highly mutated satellite DNA, which is a perfect prediction of evolution. So thank you for pointing that one out. Okay, let's have a look what else was said. You want a clear example of a functional to non-functional to functional ERV, but you don't want co-option. Well, that's how it works. It is co-option because the viral insertion is part of viral DNA, and mutations do occur. And as I've shown before, if I can show you slide number, I'm sorry, I've got that many slides and I'm forgetting where I am. I bet I'm eating all my time as well. How long have we got left?
No, I'll pause when you're looking. You do have a lot of slides, don't you?
I probably have too many. I need to
179.
179? Wow, yeah. He's prepared.
Slide 135 was the chromosome.
You got about 3 minutes left, by the way.
Yeah.
Okay, there is a paper in here. Oh, here we go. It's slide 104. And slide 105, so you can hop between these two. So slide 104, slide 105, we're talking about ERV insertions here, where we have 211 ERV insertions, of which we share 97% with chimpanzees, 98% with gorillas, 97% with orangutans, and in all of those, we have the EMV mutation, which allows us to produce a placenta. So there's an absolutely fantastic. Now, you will call that co-option maybe, or mutation over time, but that is how it works. That is absolutely how it works. The fact that we have these examples where this mutation has made something functional, and we have examples where it hasn't, is exactly what we'd expect from evolution, not from creation. Cytochrome C, we talked about last time, and you agreed last time that follows a pattern that we would predict with evolution. So that's everything you've said. What I want to say in the remaining time I've got left is if we can please go back to slide 6, please. I think you totally overlooked in your refutal the significance of what I'm showing you here. The significance of what I'm showing you here are 57 different studies taken over many, many years, independently from different people. A wide range of techniques have been applied to them, not just the same techniques. So you can't have the throwaway argument that, oh, carbon dating is false. Look, there's a bit of, it's a little bit wrong. Isochron dating doesn't work. We have a wide range of techniques that are all giving us exactly the same result. And if people were just getting things wrong, we wouldn't have 57 things telling us the same thing 57 times in such a precise pattern. This is such a specific, I feel like you really hand-wave dismissed that because obviously it's so damaging to creationism. And the reason I use this, and this is the last thing I'll say, I know you said I jumped onto the rocks and the pottery, etc. But if we are discussing is ancestry old or young, all I have to show is that that life existed over 6,000 years ago, and in terms of creation, that makes it old. And by the way, I'll remind you one more time that one of the tests that was used to get this data was examining oxygen isotope spread through the cores, use it because of marine fossils that were in there, because of life that was in there. So yeah, I think I've kind of answered everything you said, but I really want to talk more about this because I just feel like you've just walked over it and ignored it because it's so damaging. I'll be honest.
You done?
Have I still got time left? Or?
20 seconds.
That seems like a long 5 minutes.
Yeah,
I gave you a little bit extra time too, hon.
I'll call it
Okay,
dead.
perfect.
Yeah,
Bye.
I'm trying to be as equal as possible.
Awesome. I guess
All right.
since, oh, I'm sorry, go ahead.
I was just going to say open discussion now, right? Is that how you want to do it?
Sure. Yeah. I think, I guess because this time I started, maybe I can choose the first topic or ask the first question because I feel like, Catz, you kind of shrugged off the molecular clock data. I'm not sure if you're just 100% updated on it or not. For example, like the Y chromosome studies, most of them are just testing small segments. But there's only 2 studies recently, actually, that have looked at large segments. These are high-quality genetic sequences, and they show Y chromosome. Whoops! They show that Y chromosome only goes back 4,500 years. For example, it's showing that the Y chromosome mutates a lot faster than we thought, and that's why I asked you in the opening to make testable predictions, because anybody can just come up with an explanation. For example, maybe mutation rates were, slower in the past, for example. But creation scientists have actually made future testable predictions on mutation rates in people groups, for example, in Africa, where their mutation rate is not yet known. This is in the mitochondrial DNA. So my question to you is, and I'm not looking for like phylogeny-based assumptions or geology-based assumptions. Like, why do the pedigree-based studies, and I showed several, I think 4 or 5 in my opening, why when we look at the actual empirically observed mutation rate of Catz, for, you can comment on the Y chromosome, what I said too, but for the mitochondrial DNA, why do those studies indicate that mitochondrial Eve lived just recently? For example, 6,000 years ago. Even the Parsons paper, Catz, was adopted by the FBI. It was so good. So if it's good enough for the FBI, why isn't it good enough for you? So I know I asked a few questions there. So take your time, Catz.
No, it's okay. I mean, let's stick on this point for a bit and flesh it out. If I can just very quickly, Robert, just have slide 83 back. Oh, hang on. So that's the wrong one. What did I say before? It is number 122. So so on 122, I showed you here just a handful of studies on the Y chromosome, all from peer-reviewed papers, all which completely refute what you're saying. And you're telling me, okay, I get that you're telling me that there were studies out there that put it back to 4,500 years or 4,000 years or whatever. So which journals were these studies published in? Because I'm going to bet my house that they were published in creation journals and that they were totally refuted by peer-reviewed journals. Would I be right or wrong with that?
Yeah, you'd be wrong because the data was actually discovered in secular papers regarding high sequencing Y chromosomal DNA sequences. But I don't want to interrupt your time here. If you want to,
No, you're right. But the paper that made the conclusion that it dated back only 4,000 years. Okay, so it may draw on other bits of information from other papers, which is fine. But the paper that made the conclusion, it went back only 4,000 years. I am betting my house that was a creationist paper, and it was not backed by all scientific journals. Was it published? And if it was published in a proper paper, which paper was it?
Well, I guess that would be an assumption that creation journals are not proper papers, because in their journals themselves, they're making future testable predictions. Now, Dr. Nathaniel Jensen, for example, a Harvard graduate, he's looked at secular papers of high quality Y chromosomal DNA sequences, and he's discovered directly from the paper that the Y chromosome mutates a lot faster than we ever possible, for example. So he's taken that data, and here's the thing, those secular papers, of course, when they get these dates, for example, in the mitochondrial DNA and the Y chromosome that go back just 4,500 years to the Y chromosome, 6,000 years to mitochondrial DNA, when you read through those papers, now they calibrate the dates from the observed mutation rate, because there's no mutation rate slow enough in the Y chromosome and the mitochondrial DNA that can go back to 200,000 years like deep time evolution would suggest. So what they do is they assume the deep time, they use phylogeny-based assumptions. Phylogeny-based assumptions and pedigree-based studies do not corroborate. So therefore, since they're assuming the time scale, they calibrate the dates. So now they're saying, oh, the mutation rates were way slower in the past. My question to you, can you make testable prediction as to when the mutation rate in either the Y chromosome or the mitochondrial DNA slows down? When does it accelerate? Because remember, anybody can come up with a rescue device or a post hoc, ad hoc explanation. The winner of creation versus evolution comes down to who's making the best testable predictions. Dr. Nathaniel Jensen, who's looked at people groups whose mutation rates are not yet known regarding the mitochondrial DNA, he said, I predict that their mutation rate will change this many times in this many generations. This is future and novel. Evolutionists have to do the same thing if they want to have a scientific explanation. When it comes to the Y chromosome, because you would accuse it of being in a creation journal. Well, now Dr. Nathaniel Jensen, he has made specific predictions on the history of civilization regarding genetic stamps and signatures in the Y chromosome. And he's come out with a few subsequent papers that actually has detected these signatures that should not be there if deep time evolution was true. It should all just be a scrambled mess. Therefore, there's two testable predictions based on my explanations. My question to you is, okay, if these are your explanations, that mutation rates were slower in the past and we should calibrate the dates with the fossil record, what testable predictions can you make it? It doesn't have to be you. I know you're not a geneticist, but
No, I'll do it. I'll do it here now. It's not a problem.
Okay, go ahead. Yeah, go ahead. I know I talked a lot. Take your time.
You're all right, mate. I've got a couple of things I want to say. That I do want to go back to that paper with the Y chromosome. I'll only be like 2 or 3 minutes, but essentially, I think we've decided that paper you published, you talked about, was in a creationist journal, and it will have been knocked back by all scientific journals.
I want to name the paper real quick.
But you can get. I'll just finish this point, and then do definitely
take your time. Take your time.
If we just get slide 4, and the reason I mentioned that is we just get slide 43 up, please, Robert. And you mentioned Parsons as well. Now, Parsons obviously does a lot of work for the Creationist Church, and this is a paper that he was heavily involved in, and it's one Kent Hovind. Kent Hovind cites this a lot. Now, this is where he took a block of some rock from a lava dome, and he sent it off to be radiometrically tested using potassium argon analysis. Now, potassium argon dating, in my understanding, has a margin error of about 3 million years. And this piece of rock that they took come from an eruption that was 10 years ago. In fact, it does say at the bottom, I've highlighted at the bottom. And it says the laboratory was not told that the specimen was only 10 years old. So in other words, Parsons has been involved in a project where they've taken a rock, knowing that it's 10 years old, knowing that potassium argon dating gives us a 3 million year gives us a 3 million year margin of error. They've taken it somewhere to be tested, and then they've published to say, oh, look, they got it wrong because they said it was in 3 million years. If they'd have been honest and said this is only 10 years old, then the people doing that testing would have said, well, there's no point in testing that because it's well within our margin of error. You know what I mean?
Quick clarification, Catz, real quick, because what you're showing here is in June of 1992, Dr. Austin, who is Steve Austin, this has to do with the rock. Parsons' paper is a mitochondrial DNA pedigree based.
Parson was involved with this paper. I'll dig the paper out. Parson was one of the co-authors of this paper. So what I'm saying is when we're citing papers from creation journals, we really need to know what they are and what they've done, because in all the ones I've read, there are little cheap sort of tricks like this, almost like, you know, like it's a point scoring thing rather than honest science, that is why they get rejected, you know. But if you want the prediction, I'm going to take you straight back to let's have slide 10, please, Robert.
Which one?
Slide 10, please. You asked about a prediction that could be made using
molecular clock. It has to be a molecular clock, since that's what we're talking about.
Well, we're talking about is the age of species. We're talking about shared ancestry.
I just want to follow you because I don't see Parsons and anything to do with mitochondrial DNA molecular clocks in which you just, because I know what you're talking about with the RAPE project. Dr. Steve Austin is a geologist. They've revealed disconcordant dates and these different dating methods, but I don't know if we're on the same page here. Are you talking about molecular clocks, or are you talking about rocks and isotopes?
I'm just giving a scientific prediction that would only work if there was life hundreds of thousands of years ago, which has to be relevant to the discussion.
It is, but in this, I just want to narrow in. I honestly think you didn't really, because a Parsons paper that I showed in my opening, the Parsons paper is from Nature. It's not a creationist journal. It's T.J. Parsons al. Titled "A High Observed Substitution Rate in the Human Mitochondrial DNA Control Region," Nature Genetics in 1997. There's a ton of other pedigree-based mitochondrial DNA mutation rate studies. I showed several that corroborate that date, and they're not from creationist journals. And the FBI even adopted the Parsons paper. So I'm just asking a very simple question: is creation scientists are making future predictions on the rate at which mitochondrial DNA mutates in people groups that are not yet known? Evolutionists are not making those predictions. Therefore, maybe I'm wrong. Are they making these predictions? Yes. Can you make this prediction? Let's just focus right now on molecular clocks, and then we can go to your. I just don't want you to dodge around. I just kind of want to.
Okay, wait. Let him answer now. Huh? Go ahead.
So with the whole mutation rates, you are right that obviously mitochondrial DNA does mutate at a far higher rate, which is why it's useful as a tool to track migration and, you know, the out of Africa paths, etc. But again, go back to slide 83. You're telling me that you've got papers that draw conclusions, and this is the point I'm making. You've cited a paper from Nature which has talked about a mutation rate, etc. Okay, the only paper that you claim draws a conclusion that this only goes back 4,500 years is a creationist paper. You can't cite a peer-reviewed scientific paper in a respectable, reputable journal that draws the same conclusion. So I understand what you're saying. That paper has taken data about mutation rates from Nature, from whatever, you know, from proper respectable journals. But none of those journals are drawing the conclusion of 4,000 years old or 4,500 years old, whatever you said. These ones on the screen all disagree. So I don't, you know, until you can show me one that hasn't drawn the conclusion from a creationist paper. What about what? Sorry.
Well, real quick, because I showed numerous papers that corroborate with the Parsons results regarding mitochondrial leave of just 6,500 years. Why?
Can you get one on the screen and I'll look at it? Sorry, because I must have been making notes when it came up.
Sure. Yeah.
Sorry.
So let me go share screen. So my question is, if you've read the papers, I'm not sure if you have. So if you read the papers, do you know how they are? Oops. I think because you're sharing screen at the same time, I can't.
Oh, I'm not sharing screen.
Robert is.
I stopped sharing. Go ahead.
Okay, go ahead, hon.
Okay, let's see.
Oh, I had the wrong slide when I asked for 80. I should have asked 122.
Okay. So if you can see here, for example, we've got a paper here, a high observed substitution rate in the human mitochondrial DNA control region. That's not even.
You're not. Are
You're
you
not
sharing?
presenting, hon.
Oh, here, 1 sec.
And there's a little bit of a lag, so it'll be about 20 seconds
Yeah.
before I can see it. But I'll listen to what you're saying. Just don't take it off the screen until I've seen it.
If you look
Okay.
at the Catz, if you look at the meat and not the stream, you should have instantaneous results.
Yeah.
You know,
Okay,
so yeah, that sounds good.
Okay, so here we go. So if you can see it. So for example, Parsons itself is secular review. Now, I see what you're saying in that they come to that conclusion. Now, in these subsequent papers that still, you can see it here, modeling the recent common ancestry of all living humans. The most recent common ancestor of all present-day humans lived just a few thousand years ago in these models. Now, here's the Parsons paper, for
Just
example.
go back to that one. Sorry, just go back to that because I did just miss a little bit because I was a bit slow to.
So
Okay, so when it says just a few thousand years, because obviously, if a few thousand years is, 4,000 years even, then that doesn't work out because we've only got Noah getting off the ark and a handful of people. So can you tell me, and this is quite key to where this goes, can you tell me what that number is? Because just a few thousand years is very vague, and that couldn't work out well for you. What is a few thousand years?
So good question. So for one, what's good is there already is agreement between two mitochondrial mutation rate sets that exist. You want to look at multiple different studies to corroborate on the same conclusion. Okay. So for example, there's a set, the D loop, the coding region, that's germline for sure. That's given a mutation rate that is fast. You've even admitted that it's fast. And the second looks at the entire mitochondrial genome. And what's good is Dr. Nathaniel Jensen is he's taken a look at both data sets, and they both converge on the same results, the D loop, which is the control region, and the entire mitochondrial genome. So this corroborates with the Parsons paper of 6,500 years. But when you read the papers, and I'm admitting this, when you read the papers, what they do now is they consider natural selection, they consider phylogeny. They're using the phylogeny method to now take the 6,500 year date because they agree that the mutation rate in the mitochondrial DNA is fast. And same goes for the Y chromosome. Now they're calibrating the date based on their deep time assumptions. But guess what? In this debate, we are questioning the deep time. So therefore, as creationists, we're just taking the observed rate, the empirical rate based on the pedigree-based method, and we're not assuming the deep time because that's what's in question. So why would we calibrate the date with the evolutionary assumptions when the actual empirical method, the fast mitochondrial DNA mutation rate, takes us back to 6,500 years? And this is why testable predictions need to flow from this. And that's why we've made testable predictions on the Khoisan peoples. They're an African people groups whose mutation rate is not yet known. Therefore, Dr. Nathaniel Jensen, he's a Harvard graduate. He's a biologist. He said, this is how much I predict their mitochondrial DNA will change and mutate per generation. That is science. And I'm asking you, have any evolutionists actually made a prediction based on their explanation of slower mutation rates in the past? Or is that just ad hoc, post hoc? Go ahead, take your time. Can
you just go back to the slide that you had on first? And I think the important thing there that you've written, like you've highlighted the bit where it talks about all present day humans, blah, blah, blah. But the sentence afterwards that you haven't highlighted talks about all individuals that lived a few thousand years earlier than the ones that you've just highlighted. So you've basically, the sentence after the one you've highlighted just totally debunks the entire argument, because this abstract itself is talking about humans that have lived for thousands and thousands, because there's a whole cohort of people that lived a few thousand years before the ones you're even quoting.
Well, Catz, this is why I just spent a significant amount of time explaining why they believe there were ancestors before the most recent common ancestors, because they're calibrating the date now with the geological column, and they're assuming the out of Africa population bottleneck. They need to explain the low genetic diversity found in humans, which I'd like to see an answer to you from. But that's why it comes down to, and I have to emphasize this, that's why it comes down to which explanation makes the most sense scientifically. So who's making the best testable predictions? If you just admit that there are no testable predictions coming from the evolutionary community regarding their explanation for mitochondrial DNA mutation rates that are fast, back 6,500 years, then we can move on. Because I know, I've looked into it, and there's a 4-hour debate between Dr. Jensen and Dr. Venema, where Dr. Jensen challenged Dr. Venema, both qualified biologists, to make predictions on the mitochondrial DNA. And he admitted, based on the deep time evolutionary assumptions and all the factors at play, he can't. Therefore, evolutionists are not making predictions. Creation are making predictions. Our answer is science. Evolutionist answer is non-science. Go ahead.
I think the important thing is, first of all, we'll put the paper to bed. I made the point about 10 minutes ago that the creationist papers are dishonest and they cherry pick information. And we've just highlighted that, that's just been exposed like right now in front of everyone watching. That whoever's in that paper is cherry. From the abstract we've just seen, you've cited, I asked for, was there anything in any kind of peer reviewed journal? You talked about Nature, and I said, no, what will have happened is the creationist paper will have cherry picked information from Nature. And they'll draw a different conclusion. And just by looking at the abstract, we immediately see that. But what I want to move on to,
and we can move on. I don't want to
Wait. One at a time. Let Catz.
Okay, go ahead. But I don't want you to. I just want you to get the audience.
Again, one at a time, please.
Okay, go ahead.
Right, I will, because to me this is incredibly significant, and I feel like it's been dismissed in a way of just it's not important. Okay, just like, we'll just hand wave dismiss it. Do you understand that if we can get to slide 6, please, Robert, just to go back to this, and I've got so much more than just this, but it just feels like because we're skipping over it. Do you understand the significance of having 57 different investigations over tens of years by independent people using a variety of different methods that all come to the same conclusion? Do you understand why that is significant? Because what I feel that I've heard when I brought this up is you've said, there's examples of when radiocarbon dating has given the wrong result. There's examples of when isochron dating has given the wrong result. But do you understand the significance? No, it's slide number 6, please.
Sorry about that. Go ahead.
Sorry. So for slide number 6, remember, if we look at this picture, do you understand the significance of 57 independent studies using different techniques, looking at the temperature of the ocean over hundreds of years? Many of these will be using oxygen levels that have been affected by living organisms over those hundreds of thousands of years, and they're all giving us exactly the same trend, right? Using techniques that you say are flawed. Now, if they were flawed, we would have completely we wouldn't have this in sync. So what I'm asking you is, if the Earth isn't over 6,000 years old, and if there wasn't life contributing to the oxygen level testing that they were doing, why would they all be in sync like that? From 57 different tests over decades, why would they all be exactly like that? Why would they all yield the same result if those individual techniques themselves were all flawed? Sorry, I've gone on now.
Oh no, that's okay. I appreciate the response. If it got a little heated there, that's the name of the game.
I appreciate the passion. Go ahead.
Of course. And good questions there, Catz. I do appreciate the independent data. That's why when I'm looking to, for example, the Y chromosome data, the mitochondrial DNA data, the low genetic diversity data, as well as the new Rockefeller study that suggests that over 90% of all life has the same recent age due to that CO1 gene, that highly conserved mitochondrial gene that has low genetic diversity across all forms of biological life. This all corroborates, these are all independent lines of evidence that corroborate on one conclusion, that humanity and species are just a few thousand years old. And when I say a few thousand, I mean that's about 6,500 years. But this is not just post hoc ad hoc, because now we're taking this data and making testable predictions on genetic stamps found in the Y chromosome and the mitochondrial DNA, making specific predictions on speciation rates and mutation rates. I'm just going to take it as an admission that evolutionists aren't making testable predictions as to how fast or how quickly the mitochondrial DNA changes in African people groups, not yet measured. So that's fine. We can move on. So when it comes to now these methods, the same thing. You're going to say that they all corroborate, when in fact, they're always calibrating the different dates. When it comes to, like you were talking about Dr. Austin earlier, when it comes to the dating methods, potassium argon, you can look at uranium to lead, for example. Well, Dr. Austin, he's a geologist, PhD geologist, and he was part of the rate team. Okay. And these rate scientists, what they did was they took rock samples from the same rock layers. Okay. And what I'm doing is showing that these dates are often calibrated. They're not as consistent as you're making it out to be. So they take the same rocks from the same rock layers, and what they've done is they've subjected them to the dating methods all at the same time. Now, what did they find? No, they did.
It was 57 different studies over at least 20 years by different people.
Right. So these rate scientists, which are a number of PhD geologists, a geophysicist, for example, Dr. Russell Humphreys, Dr. Andrew Snelling, Dr. John Baumgartner. Dr.
Which methods are you talking about as well? Which methods?
Okay, good question. So what they're doing is they found different results for the different dating methods, including potassium argon. They've even looked at isochron dating. Now, what they found,
paper.
These are, well, for example, the discordant dates you can actually find in all the secular journals. They always admit that there are windows of error. I mean, even you've agreed that even in isochron. So, for example, now they've said, and they've made a prediction going into this project. It lasted for years. The RATE project, look it up. RATE stands for, I believe it's radioisotopes in the age of the Earth. So they made predictions. Okay, these dates must be relative and not absolute. Okay, based on all these discordant dates. So what they discovered was direct evidence in the rocks that radioisotope decay was greatly accelerated in the past. And
what evidence?
So, for example,
how do you evidence that? Because radioactive decay is you know, anyone who's done any kind of basic physics is aware of the decay constant. It's a constant for a reason. It's specific to each isotope. So what? There's only, I think, one set of conditions that can actually cause an increase, and they are extreme beyond belief that you will never find on Earth. So what conditions cause an increase in radioactive decay? But also, if you remember, when we talked about the pottery before from the Egyptian times, that gets reset every time it's exposed to daylight for less than 100 seconds. So even if you're talking about something thousands of years ago, you know, in the past, some kind of event that for some reason has
do you remember what my rebuttal was to the pottery?
Well, I don't think you, because I actually wrote down when you said that, I don't think you understand how the pottery works because I actually wrote it down.
Okay, well, I just want to answer your question for
calibration.
One at a time, guys. One at a time. Catz,
Okay. If you want to finish what you're saying on the pottery, and then I'm going to start with question 1, go to 2, and then 3. So go ahead.
So with the pottery, this isn't a technique where we measure the ratio of isotopes. You have to understand that it's nothing to do with measuring the ratio of isotopes. So if a radioactive isotope washes into a substance or washes out of a substance, it doesn't affect the result. What it does is it looks at a measure of the background radiation that's picked up over a period of time since it's been buried and hidden from the daylight. Now, once it gets exposed to the daylight, the clock resets. All right, and it might be useful while I mention this if I have slide number 18 on, please, Robert. In fact, we'll call it 19. So it gets reset every single time. Now, what this means is it is not physically possible to get a date that is older. There's no margin of error. It's just not physically possible to date some pottery using thermoluminescence to be older than it is. It's only possible to do it to make it younger than it is. Okay, it's not possible to be done, and that's why I've chosen this rather than isochron dating. So when we date stuff from the pyramids, right, which is 4,500 years old, and when we also take the boats like the Khufu boats from the pyramids, 2 of those, and we carbon date the wood, a completely different technique, and they come out to be 4,500 years old. When we date the seeds and the mummies in the different tombs, and they come out to be 4,500 years old. We're using a variety of techniques, and we're arriving at the same result. But the pottery cannot physically be dated to be older than it is. Do you understand that point? Do you understand that if pottery is dated using thermoluminescence dating to 4,000 years, it is 4,000 years or older, not 4,000 years or younger? And any contamination in this method makes it look younger. There is no type of contamination that will make this look older. It makes it look younger every single time. So therefore, if we do pottery over 4,500 years old, there were people 4,500 years ago in civilization.
Okay, so, and I'm going to address that as well. And the pottery is made from clay, correct?
Mm-hmm.
Right, we're dating minerals in the grains, different minerals. And what happens is when they're exposed to background radiation. The electrons, like I said before, they are liberated. They go into a higher energy state and they get caught in electron traps and they get stuck there. And then when we heat it, they get liberated and light is given off, and the luminescence tells us their age.
Right. Okay. So I just wanted to clarify that. So I'm gonna start right back at your first question, and then I'm gonna go right back to the pottery and the clay, for example. So the dating methods, and that's why I was pointing out that they all do have assumptions, and even isochron, for example, and I'll tell you what that assumption is. So the assumptions about initial conditions, constancy of rate, and a closed system, for example. Now, you've pointed out in the past that the assumption about a closed system and contamination does not apply to isochron dating, but isochron dating does still assume constancy, and it has an assumed history, for example.
But the contamination is reserved only for techniques that look at ratios of isotopes.
Right.
So thermoluminescent dating does not look at ratios of isotopes. So it doesn't matter what gets washed in.
I'm going to get to that one, but you asked me what evidence there is for accelerated nuclear decay. So I'm pointing out. Oh, no, that's okay. So I'm pointing out that, and you're right. So isochron dating, it analyzes 4 more samples from the same rock unit. And I'm pointing out that the rate team, which consisted of numerous PhD geologists and geophysicists, for example, they've discovered physical evidence that rapid nuclear decay has actually occurred. And the number of lines of evidence that they pointed to are helium, for example.
Citations for those, or are they creationist journals?
So, for example, like I pointed out with the disconcordant dates, those are found in all the secular journals. Now, the rate team results are found in creation-related journals with all of their sources published as well, and you're free to look at them and rebut them if you'd like. So the number of lines of evidence that they found are helium, for example, fission tracks, and radio halos. So those lines of evidence actually do suggest that the nuclear decay was accelerated during the flood. You can look at those creation papers if you're interested in rebutting that. When it comes to the helium,
but not in mainstream, no mainstream journal.
Yeah, but
has any mainstream, so it's a question really, has any mainstream journal that's not creationist ever drawn a conclusion that in history the decay constant for its specific isotope has been different? Has any journal ever done that?
Yes, secular journals even admit.
Which one?
Well, real quick, secular journals admit that heat and pressure can speed up the decay. Now, can it speed up the decay as much as is necessary to explain the millions of years dates that we get from the isochron methods and the other dating methods? No, but that's why they've gone into the rocks themselves, and the observations regarding radio halos, fission tracks, helium in zircon crystals, those are observed and documented in the secular journals. For example, like helium, as you know, it diffuses so rapidly that all of the helium should have leaked out in far less than 100,000 years. And they found rocks that are the oldest possible. I believe it was even in the Cambrian or the Precambrian. And those rocks were full of helium atoms. And the question is, why is that? And why is there so much damage in the rocks based on the radio halos, for example, and the fission tracks? Now, the point is, it's kind of like the nested hierarchies. The rule is, yes, when it comes to the dating methods, they generally come back as millions of years old. But the thing is, if accelerated nuclear decay was actually, if it took place during the Genesis flood, which was an unrepeatable, unobservable event that would have sped up decay. That's why I'm not sure if you're familiar with it. A lot of critics, they point to like the heat problem. Okay, fine. If it was accelerated, and yeah, they acknowledge the radio halos, the fission tracks, the helium, for example, the carbon that's found in the entire Earth, in diamonds as well, for example, they'll say, okay, fine. So much heat would have been produced based on that accelerated nuclear decay. Therefore, how do you explain that? So even critics that are evolutionists and from the secular journals, they admit that these observations are what are found. That means that all these dates you're pointing to, they're simply relative and not absolute. Do you want me to answer the clay?
Exact, but can I tackle the exact point you just made about heat there, first of all?
Yes,
and then I'll go back there. Okay. Now, first of all, before I tackle the heat, I just say that, you know, it's my view that there is no there's no journal other than a creations journal, and we've looked at how reliable they are, that has ever in the history of time ever said that that we've had a difference in decay rates over the history of the Earth. But having said that, something that I have recently learned, and I've just found the right slide. We're talking about massive amounts of heat. Sorry, if you give me literally 10 seconds, I will find it. I've got them all. I've got them written down. Here it is.
Well,
Okay.
can I just answer the clay one real quick?
Yeah, go on. Oh, I've got it. 67.
Okay. Go ahead, take your time.
Okay,
go ahead.
sorry, slide 67. Okay, right. So you're talking about phenomenal temperatures that are causing all sorts of all sorts of effects. So go on.
Go ahead. I just wanted you to know that we've been going for about 35 minutes, just so you guys know.
Okay.
Well, we'll make sure because I got a couple questions asked too. So why don't we finish what you're going to say about the heat? We'll finish the clay, and then if I could ask another couple questions, and then we'll jump into the Q&A.
All right. So I'll do the heat, and I'll explain the clay argument in full. Then you can take it apart and ask me the question. How about that? Does that sound all right? Okay? Or
Yeah, that's fine with me.
Cool.
I'm having fun.
So the whole thing, you're talking about these incredible temperatures that have been produced by, huge amounts of radiation, which first of all doesn't have anything to do with thermoluminescence dating of pottery, because that's just reset when it's put in the sunlight for 100 seconds. So even if that was true, it doesn't have any effect whatsoever on dating pottery. We date pottery back thousands and thousands of years, which means man is thousands of years old. So without being able to explain a link between the amount of radiation and thermoluminescence dating, then it's a non-argument. However, what you can see on the screen, I learned something recently to do with something called closure temperature. Now, closure temperature, I've highlighted on the screen, is really something to do with a temperature that if we pass that temperature, when we heat something up, we can then get diffusion of isotopes in and out of something, and we can get the contamination you're talking about. And what that effectively does is we can use. Hang on, can I go to the next? Is it on the next slide or the slide before? Right, no, it's on that slide though. We can see the bit where I've highlighted. We can talk about using really high temperatures. Where I've highlighted says artificially resetting the sample minerals. So in other words, we artificially reset the date. Okay, so if you look at something called zircon chronology, which I'm going to thank Anthony for, if you look at something called zircon chronology, so how we actually date rocks is highly relevant here. If there had been those massively high temperatures, all the age of the rocks would have been reset, because the temperatures would have passed the closure temperature, which means any kind of radioactive dating we're doing would only date the rocks back to that time. And because we don't date rocks just to 6,000 years ago or 4,000 years ago, then that could not have happened. Now, so that, and also it has no effect on thermoluminescence dating anyway with the pottery, so that's it's not an issue. But if we just go back to the pottery, and then I'll explain this in full, and then I'll shut up and you can go on. My whole argument with the pottery, because we're talking about shared ancestry and whether it's old or young, and I've always said old is obviously in creation terms over the 4,000 years, because we couldn't have had a civilization before the flood. We have here on slide 13 some pottery which was dated about 16,500 years ago, using this thermoluminescence dating, which is only going to look younger with contamination. We've talked about that. But what happens if we then look at other things also in the pyramids that have, so I go to slide 25, please, slide 25. If we look at things that are also in pyramids that have been locked away, right? All of those things, if we test them with different techniques, should come up with the same age. If they've been, but if these techniques are flawed, they won't. So here's a scientific paper for you in a credible journal, which uses thermoluminescence dating to date this at about 4,500 years old. If we go to 26 and we look at the little blue box underneath the table, you'll see about 10 different studies there. And these people, again, have independently taken samples of wood from inside the pyramid and dated it using different techniques. Obviously, radiocarbon dating is going to be a big one of them. And if we look at the one at the bottom, it puts it, again, about 4,500 years ago in exactly the same range as the thermoluminescence painting. If we skip on to the next slide, we have Khufu's ships, which I talked about, which were two ships placed in completely different parts of a pyramid when that pyramid was closed. Again, even though these were dated in complete, the first one, 1954, it was found and dated. The one in the bottom right, the second ship by a completely different team of scientists, dated using even more modern techniques. Again, both of them put about 4,500 years ago. So we've got a variety of different techniques. If we go on to slide 28, there's something here telling us about seeds that have been dated from Tutankhamun's tomb. Right, again, 4,500 years ago. So if it's flawed and we just get these random answers, why does thermoluminescence dating, why does then radiocarbon dating, why do they all support each other and give us the exact predicted results? And if they are giving us the predicted results, that means we did have civilization then, which means life didn't die in the which means life did exist civilized before the flood. And really, like the two studies I presented at the beginning, nothing is coming back other than you telling me there have been examples of contamination in the past. It's not explaining these perfect predictions and highly specific patterns and graphs. But I've had my bit now, so I'll pass on to you. Sorry.
No problem. I appreciate that. So yes, gosh, that's a lot of points. So even if I did, I'm sure you're going to disagree with my rebuttals, but let me finish what I'm saying because I need to address everything you said. So clay, from my understanding, is mostly dated using potassium to argon. And because clay is wet before it is created, let's say in an oven, for example, potassium levels range. And this is why we get different dates by different methods. So
this clay wasn't used with potassium. I know I said I wanted to interrupt, but I just want to make clear, this clay wasn't dated using potassium argon. It's dated using thermoluminescence. There's no potassium and argon. There's no contamination. So the clay I'm talking about, because you're talking about a clay that I'm not talking about. The clay I'm talking about was dated, and the papers I brought using thermoluminescent dating, not potassium argon. So
right.
You can tell me about that.
I understand there's different types of clays, but the clay is still wet in general before it's put into the oven, and then it's dried. So it's based on different types of clays that are used, but the different methods that you're saying no to that.
No, yeah, the method we take, yeah. I'm not saying that there aren't different methods we can apply to date the pot. I'm not saying that we can't use uranium, thorium, or potassium argon to date pottery. I'm not saying you can't. What I'm saying is the studies I've brought didn't. There's absolutely no study I've brought here that has used potassium argon dating or uranium thorium. So I just want to be ultra
the thermoluminescent dating, right?
Thermoluminescent dating, remember, for that, okay, does
now,
not.
But when it comes to the pottery, is that dating method giving discordant dates to the potassium argon? Well, I'm going to contest that. Yeah, I believe that there is. For example, you were saying that the heat resets rocks. Okay, but why then? And I would just claim, and I would challenge that is also an assumption, because why, when rocks date from recent volcanic eruptions, why do those come back as millions or billions of years old, for example? So that assumption based on the resetting of the clock is not really a valid assumption to begin with, because like I said, if radioisotope decay was accelerated during the Genesis flood, and I've already given a number of lines of evidence that suggest it was, then those radioisotope decay clocks could never actually be relied upon when they date rocks as millions and billions of years old. When it comes to
saying, when I say reset, and I'm sorry, and I'm only the only reason I'm interrupting now is so you understand what I've actually said rather than answer something that I've not said. That's all I'm interrupting. So once you understand what I've said, honestly, I will completely shut up. But when I talk about resetting, I am talking about thermoluminescence being reset by having a minute, 2 minutes max in the sunshine. Okay. Then when we date it using thermoluminescence again, after it's been taken indoors into a pyramid or buried, we get the date from the last time it was buried. I'm not talking about the methods that we might use to date, lava or something like that. Yeah, you can talk. I'm not saying that when I'm not saying this applies to that. What I'm saying, although obviously the lava's in the daytime, it will do. But thermoluminescence date, what the argument I'm making, specific argument.
Yeah, but you're basically saying that's an environmentally controlled. Well, real quick, man, I'm trying to be as polite as possible. In the last 20 minutes, you're cutting me off all the time. So, and the thing is, I am going to make the point that when it comes to the molecular clock dating, you weren't understanding a heck of a lot of what I was saying there. You also denied to give the predictions, but you're basically, from my understanding, you're saying that it's environmentally controlled, even when it comes to the thermal luminescent dating with the sunlight. So would we not have to know the original conditions, just like we would have to know the original conditions with all your other dating methods, potassium, argon, and the rocks, for example?
No, and that is why I've brought this. I mean, it was Anthony that told me about this, and I've done a lot of research on it, and you know, it's why I've chosen to use this to go with the argument, because honestly, okay, and I'll say it again, it can only be made to look younger, not older, through contamination. It doesn't rely on ratios of isotopes. Nothing that washes in it or washes out of it is going to change anything. The only thing that changes it is as it absorbs the background radiation. It builds up that charge. When it's exposed to the sunlight, that charge drops, and then it builds up again when it's dark, and we measure its date to the last time it was put in the dark. It is not a type of dating that you are used to. It is not a type of dating that you're going to get a half-life graph from, like with your potassium argon. It is completely different. All right, it is, and that's why I brought it, because it invalidates all those arguments that you want to make about isochron dating and potassium argon. It just doesn't apply because it's a different type of dating.
Well, I would just have to look, I guess, at the sources probably afterwards and see, because you would typically say the same thing about isochron dating, about potassium argon dating, for example, and all the other dating. Real quick, about the other dating methods, you would say, just like you did in your first debate with Ken Hovind, that, you know, the isochron dating is flawless, for example. Now I'm bringing forth arguments, objections, and evidence that actually challenges that point, and now you're looking at this thermoluminescence dating, which I'm saying and predicting that there are disconcordant dates, for example, and the original conditions would have to be known. You're saying no. Okay, I'll look at the sources afterwards, but it just sounds like you're like, oh no, you know, my isochron dating, for example, my other dating methods have been challenged. Now I'm going to go to this new dating method, and hopefully that'll rescue it all. Well, we'll just have to see if that is the fact afterwards, because I haven't looked too much into the thermoluminescence dating to begin with, but I am going to predict that, just like the other dating methods, that there's going to be some assumptions at play, and we'll just have to, I guess, sort that out at some point in the future. That's my prediction. So can I go on to a question?
Of course you can, yeah. Of course you can.
Okay. So I appreciate that. So, and I believe that was a good back and forth regarding those methods. Now, when it comes to the origin of species and separate or universal common ancestry, okay, my question would be, well, for one, I assume you're familiar with orphan genes. And if you are, you can pull up some papers, as it seems like you've got papers handy. Why do these taxonomically restricted orphan genes that are also functional to the specific organism with no evidence or sign of ancestry? You'd have specific orphan genes to, let's say, humans. You'd have specific orphan genes for some of the great apes, for example. How is this explained in the evolutionary model of universal common ancestry? Where do these orphan genes come about according to your model would be the question. So go ahead, take your time.
Right, now I did for the last debate find quite a lot of papers on orphan genes and whether, and for anyone who's not familiar with the term orphan gene, it's essentially a gene that it appears at first glance that there's no sort of detectable lineage for the gene. And essentially, I think I've summed it up really in that it appears at first glance that there's no detectable lineage for the gene. But then really, let me see if I can find the studies on it. Right, orphan genes, right, genes, Okay, let's have a look. I think I've got that on my other paper. Bloody damn it. Okay, so an orphan gene, it's something that appears to have no detectable lineage, and I will produce a Baldy Catz video on this to show that actually when the orphan genes are looked into, that actually we can detect a lineage. We can see where the mutations have happened, because obviously genetics is a relatively new science, and we are exploring the genome more and more as time goes on. And yeah.
Well, I just want to make my argument a little more clear for the argument. So, or for the audience. So the whole reasoning for the existence of the orphan genes would be based on the fact that, yeah, like you said, it appears that they pop up suddenly. Evolutionists will often claim that they popped up from non-coding regions of our genome. So they appear suddenly with no evidence for ancestry because they have no similar counterparts in other genomes. Now, the evolutionists typically that I discussed this with, they typically reject that concept of front-loaded genetic heterozygosity, for example, the created heterozygosity hypothesis that we use to explain speciation events and DNA function, for example. So they have to come up with what I believe is a rescue device, similar to the rescue devices employed for the molecular clocks. They have to come up with a rescue device to explain how these appearing cleverly designed DNA sequences have somehow popped up from the non-coding regions. Now, the answers that I've seen in the secular literature and in debates is de novo gene synthesis. Would that be, because you said you're going to make a video, would that be more or less what you would?
De novo gene seems, it to me, is just an alternative name for orphan gene. That's my understanding. It would just be an alternative name that I would use for the creation of an orphan gene.
Well, what
as it first appears.
So, well, de novo gene synthesis is pretty much the claim that, like, a non-coding region of our genome, maybe through mutation or some type of other mechanism, could pop up as a protein coding gene and become functional. And I think
which is common in our genome. That's quite common. You know, if you look at transposons, you look at ERVs that carry the promoter sequences on the long terminal repeats, we know that they can move from one part of the genome to the other, and they can carry those promoter regions with them. And those promoter regions are there to express any genetic material that's down the line. So, you know, a little bit of potluck, it might land into what might look a bit like junk DNA, but then it starts promotion, and, you know, hey ho, we've got a new protein that may or may not be functional.
I would say, just like you said, the retrotransposons, the endogenous retroviruses, the ALUs, I would say the orphan genes, they are all the same, as in that explanation of co-option of de novo gene synthesis, for example. I would challenge that and accuse it as being a circular based argument. Now, the reason why, just so you understand why I would use that argument, is because evolutionists, they say, okay, de novo gene synthesis has to be true because orphan genes exist. And why do orphan genes exist? Well, because of de novo gene synthesis. So this is a clear example of circularity, because when you look at these papers, and I've examined a number of them, they see the function. Okay, so for example, they see the function in our non-coding areas of our genome. They see the function in, let's say, introns. They see the function in the various classes of retrotransposons and the orphan genes. Now, directly in the paper, they assume that these functions had to have been co-opted, or these orphan genes must have popped up from non-coding regions of our genome. And that's why I challenged you in the first debate and the second debate to provide actual technical papers that can show certain things, like even pseudogenes, for example, because now we know that some pseudogenes produce RNAs that even increase the expression of their corresponding functional genes in the protein coding areas of our genome. So, I mean, how do you? So, here would be my question then. So if we now know, based on ENCODE, for example, accumulating papers suggesting function, how do you explain the fact that the non-protein coding RNAs that were largely assumed to be non-functional evolutionary leftovers and junk in the past, yeah, they've assumed that maybe they've got a few little functions here and there and involved in regulation, for example, but not nearly as much as ENCODE has suggested, over 80%, for example. So how do you explain that non-coding protein RNAs seem to regulate virtually all aspects of the gene expression pathway? That means, for one, it's confirmation of our model of created heterozygosity. And two, that means that the genetic degeneration that we were talking about earlier, the 100 new mutations per person per generation that are accumulating in our genomes, that means that the majority of those accumulating are indeed nearly neutral. They have very subtle effects on fitness if they're occurring in areas of our genome that are functional. So it's kind of a double whammy. It's like, whoa, these are all functional, consistent with our created genetic diversity hypothesis. And this means that our genetic information systems are degenerating a lot faster than we ever thought, because evolutionists need those junk areas so those mutations can be absorbed can be absorbed in the junk areas, making them absolutely neutral. So I know that's kind of a couple questions because I know that it's a double whammy, right? It's okay, DNA function, direct prediction, and that means genetic degeneration is faster than we ever thought. So like, how do you, and take your time, how do you address those problems?
Do tell me if I've missed the question, okay? Because I didn't write, normally I write those down, I didn't. So do tell me if I missed one and I'll come back to it. When you said, just confused me a little bit, because I did have an eye in the chat and that's really rude. And I don't know why I just got distracted, but you talked about the non-coding RNA. And when you're talking about that regulates gene expression, when you're talking about, you know, when you say non-coding RNA, you're not talking about part of the genome, you're talking about RNA molecules outside of the genome, aren't you? You're talking about epigenetics when you say that.
No. Epigenetics is a whole nother above and beyond the genome, right? Environmental factors. So for example, we know that less than 2% of the DNA codes for proteins. Now the rest, because it seems like evolutionists assume, okay, so you've got your DNA transcribed in RNA translated into protein. So they always assume, okay, that protein being the final product, that's what's functional to the genome. But no, it's so much more. Only under 2% of the DNA actually codes for functional proteins. Now the rest turns out to be like a huge controlling panel with all these millions of switches. What they're doing is they're turning on these protein genes, turning them on and off. Yeah. So
no, absolutely no. You are. I mean, gene expression is controlled and is control. You know, even like the promoter sequences, and you know, we talked about epigenetics, and there's part of the DNA that help bind those chemical compounds. So epigenetics, if you're not aware, are like compounds which regulate gene expression. So they'll regulate genes in the liver cells to make them behave like a liver cell, muscle cells to make them behave like a muscle cell, etc. and these compounds do need to bind to our DNA. So yeah, there are massive amounts. I think I know where you go. Are you going to the point that means all of our DNA is purposeful? Is that where you're going with that?
That's a good question. So as I've explained in my opening and I guess in our previous debate, a direct prediction of our created heterozygosity model that suggests that God would have front-loaded Adam and Eve with these pre-existing functional DNA differences. This would apply universally across the species as well. So now this explains speciation events. For example, this explains nested hierarchical patterns. Therefore, now we're looking to the literature. We're looking to research on DNA function and seeing if this is consistent. So yeah, I'm saying that, well, I mean, ENCODE, are you familiar with the ENCODE project? They revealed that over 80% of our genome is actively transcribed into RNA, which suggests function, suggests activity. That's a lot more than the evolutionary community have thought to be true in the past, plus the ERVs, the ALUs, the retrotransposons, the orphan genes, the introns, for example. Yeah, so I'm saying that seems to be what the evidence suggests. I mean, are you denying that? Like, how would you refute that?
This is one thing that, you know, you will find in all kind of like reputable scientific papers that, of course, there are huge amounts of non-coding regions of DNA, huge amounts of it, that do perform a function to help regulate gene expression and packaging, if you like, in the chromosomes, etc. Of course, but that is very, 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 very different to saying all of our DNA. And there is no paper out there that says that all of our DNA is useful. You know, all of our DNA serves a purpose. So we do have those, but that's almost like saying, like, if I would get into my wife's car and it's a tip and it's messy, and you know, that's like my wife pointing to the handbrake and saying, well, that's got a job, and then pointing to the mirror and saying that's got a job, and pointing to the seats and saying that's got a job, and then pointing to all the magazines she's got on the floor and the McDonald's wrappers and saying, well, therefore that must have a job also. You know, that, I know I'm using a crappy analogy there, but just because we have these bits that are well understood that regulate gene expression, you can't infer from that the bits that we know don't must have a job, because if they did have a job, we'd be finding out what that job is.
I guess I get what you're saying. If I were to just open my drawer and I found all of these socks and clothes that I haven't worn for a while, or maybe I had a treadmill that I was using all the time, and then a couple years later I didn't use it anymore, and all it's doing is sitting downstairs getting dusty and holding my clothes. I get home, throw my shirt on there or something. Someone could say, oh, that's a purpose, that's a function. So I get what you're saying, but the thing is, what's actually being discovered, like this so-called junk DNA, it's much more than that. So that analogy fails, because like your junk DNA, your non-coding DNA, for example, it actually can modify the way our packages of DNA, right? Chromosomes, for example, are organized. Therefore, it changes the way DNA functions. A better analogy is you go to a theater show, okay, and you've got all the actors and actresses on stage. Okay, what's going on behind the scenes? You got the director, you've got that person who wrote the script for the theater, you got the people controlling the lights. That's what's going on in the non-coding sections of our genome. But if you want to say that it's still the majority is junk, then I would ask you two questions. What would you predict is the max functionality of the genome? Because it was Dan Grar who looked at the ENCODE results. He said, oh no, if ENCODE is right, evolution is wrong. So he's written an extensive paper in 2017 giving a prediction and a suggestion of how functional our genome could be to keep the evolutionary story alive, kind of thing. So that would be one question. Second question would be, if it was truly junk, and I asked you this the first time, if it was truly junk, why is the cell bothering with all of this non-coding DNA, for example? Because those types of activities in the cell, going from DNA to RNA, would be very, very incredibly wasteful of energy and the resources in the cell. So natural selection then should have eliminated all of this wasteful and evolutionary leftovers long ago. So take your time if you want to answer those two questions.
My answer to that would be, slightly, I'll try and keep it quick, sorry. But why should natural selection eliminate junk DNA? You talk about why would a cell bother with it? We're talking about enzymes here. We're talking about polymerases and ligases that are stitching, copying DNA and stitching it together, and telomerases. And, they're not sentient beings. They get on that DNA like it's a zip. They zip up it, they copy it. And that's done. What does determine that is the organism as a whole. Now, if that junk DNA isn't really causing a problem, then it's neither selected for or against. If the junk DNA is causing a problem, then it causes a problem and it becomes selected out. And if it causes, if it somehow provides some advantage, if it allows, better organization of the DNA into the chromosome, if it allows us better binding of chemicals used for epigenetics, and it might get kept and used. So I mean, we're not talking about a sentient thing when we're talking about cell, and this is where it lies more in the evidence of evolution than creation, because the fact that we have these non-coding regions that we can do with or without, and they aren't in check, is completely in the opposite to creationism. But what I will say is, I feel that when we're talking about these things, we're talking about the fluff and dust. If this was a cake, maybe we're talking about the sprinkles on top of the cake, but the body of the cake itself is what I mentioned at the beginning of the debate, which is how can we have ancient things clearly made by people that have been dated using a wide variety of techniques over hundreds of thousands of years to give identical specific patterns. That's the big thing. And I feel that while we talk about junk DNA and what it could be, it's very philosophical, it's very nice, but actually it's 1% of this debate, and the first 80% was in my opening 10 minutes. We've not debunked it for sure. I think the debate was after that.
Okay, and I appreciate that, because I do feel that the one thing I didn't address is the thermoluminescent dating, and just doing a little bit of research right on the internet too. Already I've discovered a paper that says most luminescence dating methods rely on the assumption, that was my prediction, that there still are assumptions, that the mineral grains were sufficiently bleached at the time of the event being dated. So it seems like it's all based on exposure to light.
Exactly.
I'm almost done. Therefore, you would have to know that piece of pottery was either in the dark or in the light. Let's say it's sitting in a museum or underground for a while. It's going to show older because it was not exposed to the light. So like I said, just a little bit of research has shown me that no, just like you said, the isochron was infallible and your other dating methods, for example, this one also, like I said, relies on assumptions in the past, certain conditions. Go ahead. I know I spoke a lot there. Go ahead.
Doesn't. I have that slide. I have that exact slide, and I'll not bring it up because obviously you've read it and it's a paper. And if you look at it, it says that to get the date of the event. I have that exact slide. I can find it and I can bring it up and I can show everybody in the chat. But if you look at the full abstract there and the full paper, the assumption has to be made that it was fully bleached before the event of burial. That's what it means, which means if it wasn't fully bleached before burial, then it's going to date older than the burial. It doesn't mean it's not reliable. We are just dating it back to the last time it was bleached. So what I mean by that is if I take that pottery and I put it in the pyramid, I have to, if I want to date how old, how long it's been in the pyramid, which was the event, I have to make sure it's sufficiently bleached by the sunlight before I put it in the pyramid. If I don't, then I'm going to get a date from the time it was bleached before, which will be older than the time it went in the pyramid. So do you see what I'm saying? If you have the contamination, it can only make the pottery, it can't make the pottery look older than it is. If the contamination is the bleaching, that resets the clock. And if you don't have that bleaching, then it just looks as old as it would have done anyway. So I have that exact same paper, but I had the luxury of reading it all start to finish before this debate, which is why I know it's talking about the event, the burial itself. So no, it might have the word assumption, but not in the way you think. So unfortunately, that's not.
Wait. Before you get on anymore, just wanted you guys to know we got a lot of questions, and I don't want you to fall asleep or anything. You know what I'm saying? But everybody's really enjoying this, so go as long as you want.
I've had fun with this one as well. I just wanted to ask one final question regarding the point we were saying before, and then if you'd like, we can go to the next phase, which I guess would be the question and answer portion, or maybe just a minute concluding statement to wrap things up.
Absolutely.
My final statement on the thermoluminescent dating. I would just say that, and I would predict that once I look into it, especially a little bit further, that the assumptions pertaining to it are the exact same or similar to those assumptions in molecular clock dates, isochron dating, other dating methods, for example, because the molecular clocks in the mitochondrial DNA and the Y chromosome, boom, pedigree-based observed rates go back 6,000 years. Evolutionists assume their deep time history. Therefore, now they calibrate the dates with the dating methods. They assume that accelerated nuclear decay has not occurred in the past. I've given several lines of evidence suggesting that it has. There is no heat problem, for example, for a number of reasons. We can save that for another day. And yeah, sure, thermoluminescence dating, one form of dating method that I look like I'll have to look into later, but that's my prediction when it comes to that. You can comment on that, but my last question would be regarding the function of the genome. What's funny is I think I've demonstrated anyways that the vast majority seems to be active and also important in regulation, embryological development, for example, protein stagings of protein manufacture. But let's just say, hypothetically, I were to say, okay, Catz, let's just say that only 10% of the genome was functional then, okay? Because remember, it's Grar who said, if ENCODE is right, evolution is wrong. Now, here's the thing. Those 100 new mutations per person per generation that are accumulating in the human genome today, that means that 10 of them are still deleterious and harmful. And that means that even if 10% of the genome were functional, that would still lead to rapid accelerated genetic degeneration in the human line. So what type of selection? Because remember, selection is only acting upon the best beneficial mutations. And we can go over beneficial mutations, the Milano example, for example, antibiotic resistance, doesn't matter. There's a few rare ones. And it gets rid of the most detrimental of mutations. You get a mutation in one of those highly conserved mitochondrial DNA proteins, natural selection is going to remove that mutation, kill that person, for example. Natural selection is a fine-tuning mechanism, so I'm not denying that. But those nearly neutral ones, those ones that are just so slightly subtle to fitness and to the organism, they build up like a single spelling mistake in a book the size of an encyclopedia. So regardless if the genome's fully functional or only 10% functional, that problem still exists for you. So how would you explain that, Catz? And I'll give you, if you want, I'll just give you the rest of the time to answer that.
I mean, essentially, like I said before, that if creation was how things started, we wouldn't have the problem of, well, if we can call it a problem, and I really wouldn't, to be honest, but we wouldn't have the problem of these mutations building up over time. Now, we've got two possible options here. One is that if mutations build up over time throughout a population, and is that it? Does that mean a population has a limited lifespan and that's it because of that? Well, that doesn't prove creation. Or is it a case that natural selection does take its toll over time? It does eliminate those, which again isn't a prediction of creation. So either way, whichever fork in the road you take, and we could talk about the mechanisms DNA's got to correct those, but it doesn't change. There's only two paths from building up a lot of mutations. One is it ends a civilization or a population, or two, natural selection copes with it. And either one of those is not a prediction that creation would make, because creation wouldn't have that problem to start with.
Sure, of course, I'd have some things to say, but I know there's always a round in the chamber, but I don't want to open up too many.
We can have a third one.
Maybe if we just have it each a minute or two.
Yeah, absolutely. Go ahead. Go ahead.
Okay, so yeah, since I started, I guess, so my concluding statement would be I've enjoyed this discussion. I believe that the direct evidence in determining ancestry anyways is genetics. That's what's inherited, sperm and egg. And I don't believe that Catz gave any sufficient answers to the molecular clock data. He's assumed deep time, and therefore he's just kind of blindly agreed with the evolutionary and secular community that, okay, we're going to have to calibrate those dates. Funny thing is, there's no mitochondrial DNA clock or Y chromosome clock that can tick slow enough for deep time evolution to be true. And creation scientists are the ones making the testable predictions. When it comes to the indirect lines of evidence, like the dating methods, I've demonstrated that they all rely on different assumptions about initial conditions, constancy of rate, the fact of a closed system, and even isochron dating will assume that there was no accelerated nuclear decay in the past, which I've given plenty of evidence for directly in the rocks themselves, even with the rate team. Now, when it comes to the thermoluminescence dating, I've given a couple responses based on the limited knowledge that I even know about that dating method. I'll look more into it, but I'm predicting that it's the same as all your other dating methods, for example. So when it comes to the function of the genome, evolution has always assumed that the majority is based on evolutionary leftovers, and that should reflect those nested hierarchical patterns. So I just want Catz to understand that no, if we both predict nested hierarchical patterns, okay, in morphology, anatomy, DNA, and physiology, that means DNA function can actually help differentiate between the two, universal ancestry or separate ancestry. And it's my point that the data and the most recent literature has suggested that the vast majority of our genome is functional, which is a double whammy. It's a fulfilled prediction from the created heterozygosity hypothesis, and it's also it's a problem for evolution, because that means that we are degenerating a lot faster than we ever could. That means the time frame based on mutation accumulation can't be deep time, because those mutations would have driven us to extinction long ago and far away. So, but overall, I've really enjoyed the discussion. We've touched on a number of things. Time has flown by. It's already been a couple hours. So yeah, that's all I'd have to say for my closing. So Catz, take it away.
Well, I've enjoyed the discussion as well. I'll be a little bit quicker. I think I've only got two things to say. I think the discussion about genetics is great. Neither of us are professional geneticists, and there'll be people in the chat that know more about the topic than both you and I. But I feel talking about, us sat here talking about the genetics and the gene regulation, etc., is a bit like us talking about what we're going to buy a girl, for a meal tonight before we've even asked her out. It's a bit premature, because the big thing is what comes before that, right? If we're going to separate evolution from creation, then the big thing is creation says 4,000 years ago there was a flood and death happened after that. The population grew from then. And there is such an abundance of evidence that there were civilizations and life before 4,000 and 6,000 years ago, that if we look at that evidence honestly, then creation doesn't even start. It doesn't even get to the point where we start that. And I've shown 3 studies tonight. I just want to recap. I've shown one where we have 57 independent studies that have been taken over a period of a couple of decades. I'm sure Anthony will go into that in way better detail than I could when we
saw him. Okay.
Yeah, I've got it. He will go into way better detail than I ever could. And I just hope I've not let him down tonight in some of my explanations. But we've shown that study that uses a variety of techniques to produce an astonishingly identical results in the graph. We saw the two Antarctic ice cores that again were dated independently by independent people miles apart, which then is corroborated by an ice core which is dated I'm looking at the volume of ice over hundreds of thousands of years with completely different technique again. Complete different techniques, giving astonishingly similar results. We talked about thermoluminescent dating and how that can only make things look younger, not older, right? But that puts this pottery in the pyramids at 4,500 years old. We looked at a study where people looked at wood found in the pyramids, which had about 12 different people all doing their own dating, which all put that at 4,500 years old. We looked at the two Khufu ships, and I know I've pronounced that wrong, sorry, which again, 40 years apart, with different techniques have been dated to be 4,500 years old. So really, I cannot buy into the fact that our dating techniques are not correct when they all give the same result. And if they're giving the same result, yes, you might go and find one fly in the ointment and say, oh, but this, but I tell you what, science is the first to call them out. I've got one slide that I've not shown, which looks at uranium thorium dating, which puts a cave painting at 65,000 years old. And it was called out by scientists in a refutable paper because they said that the water that was running over the stalactite they'd used to date it had, I think it had washed out some uranium, and they called it out because science regulates itself. Science spots the errors. If these flaws were there, science spots it. They regulate themselves. They rein themselves in because they call each other out. There's no agenda from scientists to pretend. So, I think when we start looking at the data, honestly, there is no argument because creation can't even start. But that's my finishing touch.
That was so cool. You guys are so good at this. I say, everybody's really enjoyed it.
I've had a blast, and I would do a round 3 because Standing for Truth is such a nice guy to debate. And I know a couple of times we got a bit passionate with it, but I don't think there was ever any malice, and I would happily make this into a series of debates.
I think that's a good idea. I've had a good time. It always gets a little passionate at times, but that just shows we're, that invested and interested in the topic. So I've had a blast, and I'd be more than happy to further it out in a third round someday.
You're both welcome anytime. And I got most of these are for Standing for Truth. So, but you're both welcome to
usually
say something. Yeah. You have had a lot of supporters here, though, Standing for Truth, and I'm happy to see that. I'm so happy to see that.
Don't you forget to answer. Don't you forget to ask my question for Catz.
Shut up.
Now, I do have a request, and of course, let's get through as many questions as possible.
Go ahead.
In my, it's after 8 where I am. I got to put the kids to bed soon, but I'm not saying maybe just a little bit of a time limit on the question.
Absolutely. Right.
Okay.
And as you see, Tony has joined us, Anthony Purcell. All right. So the first one's from Dave Holmes. It's for Standing for Truth, of course. So if Earth is young, please explain 500-foot chalk layers.
If the Earth is young, please explain 500-foot chalk layers. Real quick, I guess those would have been formed during the global flood chemically, all limestone. And Catz is probably aware of this. And contains the calcium carbonate, not from coral like the uniformitarian assumption. So that would just be a flood-related occurrence. Actually, that would be a good topic for a third debate, would be the global flood and some of the effects on that. But yeah, that's what I got.
I'd love that. As I said before, I'd love that to be a topic. Catz, do you want to say anything about that one, hon? Or Anthony can, I guess, join too.
I was just going to type. I can just hear the little one stirring. Can I just have 30 seconds check on it?
Absolutely. Yeah, no. Anthony, you want to take his place? Anthony? No? Okay. I'll go on. All right. I think I saw one of the super chats I did.
Babe, Anthony's muted. I don't think he...
Yeah, Anthony, you're muted.
Sorry,
okay.
I was muted there. So I would suggest that there are a wide variety of very slow geochemical and geophysical processes that are simply inconsistent. For instance, there are stalactites that are many hundreds of thousands of years old. So the assertion that the Earth is 4,700 years old is completely inconsistent with geophysical evidence.
Well, I guess that this is my question. I'll have the last word. So,
Yeah, absolutely.
even going into the accelerated nuclear decay and the changing conditions in the past due to the global flood, we can go over all the countless of geological related features that show that a global flood has occurred. I mean, we see flat featureless boundaries, we see rapid or no erosion between sediment layers, but it always comes down to, as I say, Oh, no, I didn't interrupt you. It always comes down to that which can make testable predictions. Now, one of the predictions that has been confirmed that I'll just go over with real quick regarding the catastrophic plate tectonics model, meters per second plate movements. If these plates are being pushed down, as it's suggested by the flood geology model, and haven't had time to melt, we would predict that there should exist massive slabs of cold rock at the base of the mantle. Dr. John Baumgartner has actually made this prediction, and years later, a new field of science called seismic tomography, they've actually learned that these massive slabs of cool rock exist at the base of the mantle. That means they haven't been pushed down long ago because they would have melted. They've only just been pushed down recently. An amazing confirmation of the global flood model, a testable prediction that's been confirmed. So that's what it comes down to. Is
errant nonsense.
We gotta move on, though. We gotta move on.
And this
we
is
have lots of questions.
debate, so if Anthony wants to set up,
I really would like to respond to that.
Wait, Anthony, I would love. We can have to do it next time.
All right, guys.
Sorry, but I got so many, and they got to get going because it's really late over there. All right, so, this one's to Catz actually, but it's from Dapper Dino. So is he jumping sides? Assuming radioisotope decay could be significantly accelerated, would that affect electron resonance dating or of the pottery? Why or why not?
Right now, I'm sure Anthony could answer this a lot better than I am. So we'll see if he gives me a thumbs up or thumbs down. But no, look, like I said before, with the pottery, people have tried to fake pottery to make it look more expensive than it actually, make it look older, to make it look more expensive. And with the slide I showed before, it is detectable. So if you have had some amazing sort of like short spell of high level radiation, then you will have normally the grains of different sizes will have a different buildup of charge in them. And that difference is affected noticeably if you have a massive dose of radiation in one go, and that's how they detect forgeries, according to my research. But all of that is irrelevant anyway, because let's say we had a couple of thousand years ago, amazing high radiation, and I don't know why. That's instantly bleached out. If I've got this piece of pottery that's out in the daylight before it's buried in the pyramid, then it's bleached out within 100 seconds. 100 seconds of daylight is all it takes. So, no, there's the answer to that.
Go ahead, Standing for Truth.
Yeah, I would just say that that would assume that it's like what we see in the lab, oftentimes, or even what the secularists agree occurs, that heat and pressure can speed up decay rates. But when you're talking about something like the global flood and the accelerated nuclear decay that occurred at that time, combined with the lines of evidence that the RATE project has discovered, for example, helium, fission tracks, and radio halos, which I don't even think Catz attempted to address, then that would pretty much speed up. Well, for one, it's going to make these dates, as I've said, regarding the rocks, the pottery, it will make them relative as compared to absolute. So that's just a few problems regarding
May I make a statement at this point?
That Catz would have to discuss possibly in a third debate, because we didn't really even get to touch on the carbon-14. The evidence is there. So, but that's just my prediction, that those assumptions also apply to the thermoluminescence dating. So it was Catz's question, so he can have the last word. That's only fair.
How about, sir, I'd like to make 2 points. When you're
this isn't a 2-on-1 debate. I don't know what's going on. How about I call one of my
Yes,
So-
why don't you?
No.
Instead of interrupting me?
Well, no, wait. I brought Anthony on for after the debate just to talk because he's a friend of everybody. I didn't expect it to get heated. We don't do that.
I apologize.
No
I would
problem.
suggest- I
So
would suggest once this
I-
debate's over. No, I'm not, because you know what? Him and I just had a back and forth on the global flood. We're not going to have a back and forth on every single question.
No.
okay, can I make a statement though?
want to-
I am a professional geophysicist, and you are making assertions about the state of geophysical research that are simply false.
Gary.
Okay.
All right, hey, I would like to have you both back if you want to do that, Anthony. Would you like to come back and just have
Sure,
it on with him?
absolutely. If you
All right,
want
why don't
me,
we do that? Why don't we do it
and
that way
why
then?
don't I just
Yeah, I'm
mute or
happy.
leave?
I mean,
Okay.
I debate almost weekly. I'm not going to have a second
No.
debate here in the question and answer.
No problem. Okay. So, oh boy. Next one would be from creationist myths for standing for truth, of course. Can you name a specific? All right, I'm gonna mess this up, guys. Sorry. Transposon, retrotransposon, or ERB that has a documented. What did I write? I didn't write it down right. I'm sorry, guys. I'll read another one until I get that better. From TD Lane for standing for truth. How is? Oh God. Why do you guys use big words?
You're doing
is
a great job, Marvel girl.
heterozygosity? Not
That's
an untestable
Sounds good to
That's
me.
it.
assumption.
That.
Woo! Yes, not an untestable assumption.
What did he say?
How is heterozygosity not an untestable assumption?
How is it? Well, it is testable, and this is especially someone like T.D. Lane. I mean, he's heard me discuss this many times. I've suggested that if God really did front load Adam and Eve and then the created kinds with these pre-existing functional DNA differences, this means that the testable predictions that would flow from it, and they've been made, they're on paper, pertain to DNA function, pertains to DNA function in these highly conserved mitochondrial DNA proteins, for example, the cytochrome C. So there's also predictions on mutation rates and speciation rates. I invite him to read the technical papers that go over the predictions in great detail. So that's why I often focus on function, because if the vast majority of our genome really is junk and based on evolutionary leftovers and genomic fossils and ancient infections of parasites and viruses, then I would concede that that would be a falsification of that hypothesis. But the evidence seems to suggest that the vast majority of our genome is active. So I invite T.D. Lane to make a prediction on DNA function and attempt to falsify it. That's how science is done. So good question.
Catz, do you want to say anything about what is heterozygosity? If you want to tell me.
Good question. It's a state of genetic differences, millions of DNA differences in chromosome pairs. It suggests that genetically it goes back to your alleles. You know, you've got capital A, lowercase a, capital B, lowercase b. If an allele is capital A, lowercase a, that allele is that gene is heterozygous, for example. So it gets a little technical, but
I knew that.
The creation argument to that is that when when Noah got off the ark with that small population he was on the ark with, they contained within them all the alleles that have produced all the variation which exists now, which is just it's mathematically totally impossible. So
I was wondering about how they talked about the population of the Earth from the very few people. When it comes to the scientific, the alleles and everything, like you said.
Well,
if you want to say something, go ahead.
A couple today could hypothetically have millions and millions of children because they're obviously not because they're not gonna live long enough. But we have heterozygous gene sites in our genome. That potential, for example, me and Catz have about 3 million places, more or less, about 3 million places where the copy we got from our mother differs from the copy that we got from our father. Now, if we carry just me and cats, for example, 3 million differences. It's not a huge stretch to take the DNA differences, the SNPs found around the world, take those 10 million and throw them into Adam. Therefore, Adam and Eve have the potential to produce every shade of skin that exists on the planet. So it's scientific and the predictions have been made. So I invite anybody to make better predictions and falsify those predictions. So.
And do you think, I'm sorry to get away from their questions, but I wondered about this. So what about the numbers of the people? Like, do you think it really, don't creationists say it like doubled and doubled or something like that?
Yeah, I mean, if you actually take back the number of people we have today, once you start going back far enough, it's really hard to track genealogies because then we're all so closely related. For example, there's been so much intermixing, interbreeding, but the population growth rate really does just go back to 4,500 years. Evolutionists oftentimes have to invoke extinction events or plagues or other types of factors to account for their date of 200,000 years for the out of Africa. But yeah, the population fits quite nicely with the young Earth creation timescale.
What about those extinction events in the creationist model?
Yeah, there would have been, well, for example, there would have been a near extinction event with the flood. That would have been a population bottleneck. The creation model has essentially 3 bottlenecks. You got a bottleneck at creation with Adam and Eve, a bottleneck at the flood with 8 people, and then a brief bottleneck at the Babel. The thing with the creation model bottlenecks, though, is they were all one generation and they were followed by rapid and exponential population growth. Therefore, there wouldn't be any inbreeding problems. There wouldn't be any significant loss of that original created heterozygosity. Now, if you get into the evolutionary population bottleneck, which was extended for over 1,000 years within a population of 2 to 10,000, now there would have been some significant inbreeding problems, which would result in rapid genetic degeneration. So bottlenecks and extinction events are a problem for the evolutionists. And unfortunately, Catz and I didn't get to discuss that too much, but I did discuss it in my opening. So good question.
Yeah. Catz, do you have anything you want to say about that?
No, I think when we talk about inbreeding, I don't understand how you can address a bottleneck and say that's going to cause inbreeding problems when you claim that you've got 3 people stepping off the ark and producing an entire population.
I'm getting dressed.
Yeah,
they're wrapping up.
Oh, we're wrapping up.
Are you talking to
I'm
us?
sorry,
Robert?
guys. I thought that I was muted. I'm so sorry.
Okay, no problem, babe. No problem.
Who are you yelling at?
I was like, what?
So,
All right,
yeah,
go ahead, Catz.
I think if we're going to talk about inbreeding, that's going to happen because, like I said before, it's totally impossible for a small number of people stepping off one boat to have all of the variation, all the alleles needed for the variation today. And if they did start to breed, the inbreeding would be more severe than anything we've seen anywhere. But then it only takes one bacterial cell, to produce an entire colony. So, who knows? But how much variation is there in that colony? Not a great deal. And of course, they were produced by a different method anyway. So I don't know why I'm waffling on about that now. But yeah, no, it's nonsense. The inbreeding that will be caused by 2 or 3 people stepping off an ark is ridiculous.
Well, just to respond to that real quick, I guess, because it is an interesting point that you made. I do want to emphasize that according to our model, the 7 billion people on the planet today descend from ultimately 3 founding couples, Noah's 3 daughters-in-law and Noah's 3 sons, for example. Fascinatingly, we see 3 main mitochondrial DNA lines around the globe on every single continent, L, M, and N. So that evolutionists will just say that's a coincidence, but it's fascinating confirmation of that model. But that's more than possible because we don't explain the vast majority of DNA differences as a result of mutations. And I feel like I got to say that over and over again. We explain the vast majority of DNA differences as a result of created heterozygosity. That means there would be no inbreeding problem because evolutionists explain the vast majority of DNA differences as the result of mutation. So by the time you reach the out of Africa scenario, you've got the Australopithecines evolving into Homo erectus. Homo erectus evolves into Homo sapiens, for example, or Homo sapiens. There's been so many generations of mutation accumulation that by the time you reach that bottleneck, now you've got all these recessive mutations that are coming to the forefront, leading to rapid genetic degeneration. But inbreeding is the exposing of the hidden reservoir of genetic mistakes. In the creation model, up to the flood, there wouldn't be any accumulation. There'd be a few, but very little accumulation of genetic mutations and genetic mistakes. So if there's no mistakes to lead to a significant inbreeding problem, there wouldn't be an inbreeding problem like there would be with the evolutionary bottleneck. But if those DNA differences are front-loaded from the start, then therefore processes like recombination, gene conversion, shuffle around those DNA differences could produce variety rapidly, even in a single generation. That goes for the archines as well. So it's a model that leads to testable predictions, and that's what needs to be addressed, is the predictions. That
that's and I know this is a question thing. I'll just say one thing on that, and then I'll leave it. Back in sort of like, testable prediction, where do we see in breeding? We look at selective breeding, don't we? We look at selective breeding, which is what humans and farmers have been doing for hundreds and hundreds of years. And with selective breeding, which is a form of inbreeding, we breed out the variation, and we get similar. We don't breed in variation. We don't have 7 people step off an ark, inbreed, and create variation. Selective breeding, where we have a small number of organisms and we are breeding them together, it reduces variation over time. So it's not just about accumulated mutations, which can then come to the front. It's about a reduction in variation. Going out and meeting other people who are very different to you, and that's how variation is spread, as well as mutation. Inbreeding will reduce it.
That's actually a good point, because as creationists, since we believe in the created heterozygosity hypothesis, we look back to the creation event and we see the expansion of the genome, because these changes, these speciation events, have resulted from shifts in heterozygosity to homozygosity, which means reduction in variation. That's why when we look to species today, for example, we can look at the 7 living equid species. They have more homogeneous gene sites than they do heterozygous, which makes sense according to our model, because we're looking at reduction in genetic variation. You can see that with 2 wolves. You can see that with dog breeding. For example, 2 Chihuahuas are not going to have very much allelic variation. They're not going to have much genetic potential. And what I find funny, and this will be my last point, is, like I said, with the equid species, you've got 7 altogether. I believe it's 3 species of zebra, 1 wild horse species, 3 wild asses. Now, when it comes to the breeds, we've got over 850 breeds of horses and donkeys in the world today. And the evolutionists will even admit that these breeds have come about in human history, because they've been created through the recombination, for example, shuffling around those DNA differences, resulting in new variation. This has been done by humans. Now, evolutionists will say that those 7 little itty bitty species of equids took, I believe it's 4 million years for those 7 to come about, but they will even agree that it only took thousands of years for the 700 plus breeds to come about. But then they think there's a huge problem
at the risk of triggering you. You have spoken for a long time. You seem to like to dominate the rhythm of conversations by just waffling about stuff that is peripherally related. Please keep your answers succinct. You're the person who claimed to be operating under a time limit. Oh,
wow. Okay. I wanted to ask another question, because a few people have asked about this. It's a study that they want to know if you've seen it. Standing for Truth.
Of course.
2017 UF study: snail kite bird evolves virtually overnight to keep up with invasive prey in Gainesville, Florida. Have you seen that?
It's speciated literally overnight, is what it said.
It's evolved virtually overnight. I don't know the study, so I don't know what it says. It's from TP Seeker. If he's still here, he can let us know. And I think TD Lane also mentioned it.
It's very interesting, because we've got future testable predictions on speciation rates. We look at, for example, there's only about 10 to 12,000 bird species. We take that back 4,500 years to the flood. That's roughly 2 to 4 new bird species a year. Therefore, we should be expecting to see observed new species of bird in real time. And just last year, it was a paper came out where there's been a new species of Darwin finches on the Galapagos Islands in real time. And what's funny is the speciation event resulted from shifts from heterozygosity to homozygosity just has been predicted based on this created heterozygosity hypothesis. So what I find funny, and if Catz wants to respond, he can, evolution would say that birds evolved from theropod dinosaurs roughly 65 million years ago.
Right.
Why is there so few bird species today? 10 to 12,000 species. This also goes for lizard species, snake species. There's only a few thousand species per like creature, for example. So yeah, go ahead. You can have a word there if you want, Catz.
Well, just on speciation, I know you were talking about birds, and I know insects didn't go on the ark, but there are millions, and I presented this argument to Kent Hovind, there are millions and millions of species of insect, and we will be needing, you know, I think I forgot my slide here, 3 or 4 new species of insect a week. So yeah, you know, you might be able to find one species that fits, and that's based on 6,000 years, and that was just using flies, wasps, fleas, bees, bee kind, because I included bees and wasps, and aphids. You know, that was just using those as an example in the calculation, which I can put in a video if you want. But yeah, when we look at insects, you know, yeah, we're looking at almost on a daily basis. We should be seeing new insects, which we just don't see. So yeah, the speciation rate is way too high.
How come there's so few bird species? Like I said, if birds evolved from theropod-like dinosaurs 65 million years ago, why do the number of bird species between 10 and 12,000 correlate so well with the young Earth creation?
2 words: extinction rate.
Right, I knew you were going to say that. What kind of predictions can you make on extinction events? How many extinction events? How fast are these speciation rates? We've observed in real time last year a new species of bird. The number of species, insects, there's only a 1 million species of insects. That includes everything that you've explained. Insects survive a flood, no problem. Insects can just make their way onto the ark, no problem. That's easy. But when it comes to like mammals, reptiles, and birds, I'm talking here. Relax.
Don't interrupt. Don't interrupt.
34,000 species. For example, lizards and snakes. I don't have the exact data here. There's only about for bats, there's 1,100 species. Lizards, I believe there's 4,000. Snakes, there's about 4,000. That's one new species per year. The species that we see do not correlate with deep time evolution. They correlate much better with young Earth creation timescale. And Anthony here just proved his non-science and his post hoc, ad hoc, ridiculous rescue devices because extinction events. Show me some testable predictions on extinction events or that's just non-science and rescue device. Go ahead.
Right. I mean, look, at the end of the day, you've picked a species there, you picked a bird, and that seems to fit your thing. Ultimately, it just doesn't. I mean, I presented this argument in a 20-minute part of my debate with Kent Hovind, and you know, there's more. I could talk about rabbits. I could talk about rabbit kind, beetle kind. We need a new species of rabbit every week. Species of rabbit. So let me just go back there. Now that depends whether you want to talk about rabbit or rabbit and hare kind. We need to see a new species of rabbit every 2 years, and we need to see
breeds or species.
Say that again, sorry.
Is that a combination of species and breeds, or just species?
We're talking about species, actual different species, not breeds and species.
Okay,
and that's
I looked up your slide after, and you accidentally included breeds in your with rabbit species. Once again, there's only a few that correlate nicely with the young Earth creation timescale. You can't include that. Would be like me including the equid species, the living 7 in the wild, with the 850 breeds. Breeds are easy to come about. Humans do it naturally. You got to look at that which is in the wild, Catz. So you might.
How do you define a breed? How do you define a because when I'm talking about go on. How do you define a breed?
Definitions are
about artificial selective means from humans. So obviously they came about during human history. So the rabbit species in the wild, there's only a few. There's only a few species of rabbits. There's only a few species of birds, lizards, snakes. Every animal you can point to fits well with creation.
Wait a minute. Are you saying? Excuse me. I'm sorry. I'm trying to follow. I'm just trying to follow, babe. So are you saying that humans make breeds of everything? Like, are we? What?
by definition, breed versus species, okay? There's a difference between like your Canis coyotes, Canis familiaris, you know, all your different species of dogs versus your 450 breeds. The breeds have been artificially selected from humans. They've done the job. That's, you know, just through reshuffling of these genetic DNA differences. The species in the wild, those are the ones that we need to explain how they came from the flood.
Right. I thought you were talking about every animal, not just dogs.
It wouldn't make any sense when you're saying, oh, you need over 300 rabbit species. No, because most of those are breeds. Those came about from human means anyways.
Oh,
It's
okay.
just the species.
And you add to that, because you talk about kinds coming off the ark. So it wouldn't just be the rabbits. It will be the rabbits and her, because that will be the same kind as defined by Kent Hovind. And then if you talk about kinds, you add the pica to that as well. And that's what I was doing. You know, so unless you want to talk about rabbits as being one animal, hers being another, pica being another, in which case they are all on the ark together. And if we start doing that with animals, then suddenly the ark becomes too full because no ship can carry them. So when we're talking about the speciation from a kind, in which case the speciation rate is unbelievable. Or are we talking about speciation from all the animals we've got anyway, you know, which means, do you know what I mean? Like, which means
when it comes to, by rule, by definition, species and breeds, there's always more. There might be a few cases, but you know, the majority proves the rule. There's always more breeds than there is species. The species are very limited. Now, when you look to this idea, it's very simple. When you look to this idea of pre-existing diversity, okay? So for example, let's take the cat ancestor aboard the ark. There's only about 37 cat species in the wild, okay? So say that this cat ancestor was front-loaded with a whole bunch of functional DNA differences at creation. Now, these front-loaded functional DNA differences has led to the origin of species. For example, Noah brings 2 cats. Now we've got everything from tigers to house cats to jaguars and in between. If you look to dogs, same thing applies. You've got everything from wolves to coyotes to jackals and foxes.
You're talking about the cat. You're not talking about taking species on there. You're talking about taking kinds on the
right.
ark.
So Noah would have taken, let's say Noah took 2 bears. Now, those bears would have originally been front-loaded with all these functional DNA differences. It's led to the origin of species. Now we have black bears, brown bears, polar bears, and as a matter of fact, all 8 species of bear. Like 8 species of bear is not hard to explain, is my point.
What I'm saying, if you take kinds on the ark, then the speciation rate has to shoot up rather than taking species. And I mean, again, it's something maybe I'll put in another video, but you know, I will politely say that you are just massively wrong with that. The speciation rate, if you're taking kinds, is way. I mean, there are 350,000 different species of beetle alone. Now that means that we need 1 species of beetle.
You keep going to insects and avoiding mammals and reptiles and amphibians. Why is there only 8 species of bear? Why is there only 37 species of cats?
All right, but your model should have to fit to every single organism. Like if we 350,000 species of beetle and we took 1 kind on the ark, then, and I'm just looking at my slide now that I used, that calculated over 6,000 years, if we didn't put them on the ark because they're an insect, we need a new species of beetle every single week. And we're not getting new species.
Bees so when bees live a few months, a queen lays thousands upon thousands of eggs. There's only 1 million species of insects. Like I said, they can survive a flood easily. They can survive on log mats. They can survive in the ark just by going there on accident. When we focus on the 34,000 mammals, reptiles, amphibians, birds, there's so few for deep time evolution to be true. That's why, just like Anthony proved to us here, you have to resort to a rescue device of extinction events. No, all we do is linear speciation events since the flood 4,500 years ago easily explains the number of bird species, reptile species, mammal species. I just showed that it's consistent with your cat species, your bear species, your dog species. We're the ones making the predictions. Evolutionists can't.
Are you claiming?
Wait.
I am going to interrupt you.
No, come on.
No, because he does this all the time. He will talk for 5 minutes about nothing much.
Anthony, you're just
repeating himself,
crashing,
repeating himself.
You should leave it to Catz. He's a better debater than you. You said
come on. No, we're all going to be friendly.
So,
be friendly.
Prediction on the number of
make me turn this car around.
I'm having fun here, but it's going on 3 hours. We're gonna have to.
Yeah, we gotta call it, guys, because everybody's gotta go at this point.
It's been fun. It's been fun.
It has been, and I really appreciate you guys. Now, I want everybody to take Does anybody want to say anything before they leave?
I just want to say it's been fun. I mean, we probably could do this all night. It's been a few hours.
Yes,
We've touched on
yes.
a lot of topics. If it ever got slightly heated with Catz and I don't think it has, but I would apologize because I enjoy debates and discussion with Catz. I'm just not into like a 2-on-1 in the
No,
Q&A
of course not. And that was my mistake. My mistake. My mistake. So,
No, you're a great host.
Catz, would you like to say anything before we?
Yeah, I enjoyed it. I mean, you know, I think ultimately everybody watching is going to make the mind up over who brought, you know, the evidence and citations and who did that best. You know, so let's see what the comments are in the comment section afterwards. But I enjoyed it, and I'll be up for round 3.
Yeah, I got a million questions. I could ask them all night, but we're gonna have to do this some other time because you guys need to go deal with your kids, and if there might be an after show, I don't know. People are asking, and thank you everybody for the super chats, and we'll talk to you soon. If there's an after show, it'll be in my Discord. I won't stream that one. Okay.
Now, can I make one request before we end?
Absolutely.
Can Tony give me a smile?
Please smile for us,
so
Tony.
sad right now.
Tony's a sweetheart. Tony's a sweetheart.
I am upset by the degree of intellectual dishonesty.
Well, your world is
display in this debate.
I'd be upset too.
Let's leave this on a high note.
Let's leave this on a
high note.
We got it.
Don't make me yell, because I'm Italian. I'm louder than you.
What I will say is, though, like what I do want to say is, I think I said it right at the very beginning of the debate. That, you know, I enjoy learning new stuff. You know.
Oh yeah. Yeah.
And you know, and since meeting Tony and keeping in touch with him after he was on my channel, honestly, standing truth, I have been a very pale imitation in terms of what I was able to present and how I could present it to what Tony could do, you know. And I would love to see the two of you talk sometime because, you know, while I might fumbled over my words and tried to recall studies I'm not familiar with, and so, you know, Tony knows his stuff inside out. He's probably forgotten just in the last 20 minutes more than I've learned this entire debate, you know. So it would be very interesting to see you talk. I've got a lot of respect for everything, and I just hope that I didn't cock things up too much from everything.
No, you didn't. You did a fantastic job, Catz.
You did a good job. It's been fun.
It was great. I loved it.
Thermoluminescent dating, you know, and that's the point. Is I like. It's not every debate where something's brought up to me where I'm like, this one I'm gonna have to look a little more into, and that's what I look for. I mean, it gets stale if it's just the same thing every time. So no, you brought forth some good arguments, and I've enjoyed them all one by one.
I have to say, but go ahead, Tony. Then I'll say something.
Well, if you're going to do some research, I suggest you look up cosmogenic radionuclide dating, amino acid racemization dating, VAV chronology, dendrochronology, and rhythmites.
That snail bird. But I think
real quick,
things like that you catch in these types of debates, not the yelling debates. Go ahead, Robert Hun.
I have to go, everyone. Thank you all for coming and debating, and thanks to the audience and everybody who shows up week in and week out. We appreciate you all, and I'll see you on the next stream. But
all right, take care, Robert. See ya.
Thank you, Robert. Yeah, he's got to go.
Thanks, Rob.
But yeah, so yeah, I think we catch things more in these debates than when we're yelling at each other and calling each other names.
I will also apologize because I have a very different presence than Catz does. Catz is very quiet. If you start a discussion point from what I regard as a false premise, then I won't let you finish it. Because from a false premise, you can end up wherever you want. So I am a much more egregious interrupter than Catz is. Catz is a much more polite and restrained person than I am.
But we try our best around here. We do. Okay, so thank you very much. If you want to come over to my Discord afterwards, I'll be around to see if anybody shows up. If not, good night, everybody.
Good night. Thanks again. It's been a blast.
Bye-bye.
Good night, everybody.
I got it, babe. I'm still here.
Okay, good. Thank you.